Good morning, everyone. Great to see a great crowd here, 8:30 in the morning. Thanks, everybody, for turning up. This is Gairdner Science Week, and this is one of the highlights of the week. This is the Laureate Lectures, where we're going to hear from all of this year's Gairdner Award Laureates and their great work. I'm Janet Rossand. I'm President and Scientific Director of the Gairdner Foundation, and I'm very happy to welcome you all here in person. And we also have a lot of people online in Toronto and around the world, so this is a great event. Before we begin, I'd like to take a moment to acknowledge that Mars, where we're at, is located in Toronto on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis people. This territory is subject to the Dish of One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. This event, as I said at the beginning, is part of Gairdner Science Week, which is really an annual week-long celebration of our Gairdner Award Laureates and the impact of their research on human health. And I'd like to thank our long-term Gairdner partners, the Government of Canada through CIHR, the Government of Quebec, and the Government of Alberta. And a special thanks to all of our Gairdner Science Week sponsors, up on the slide here, who've made these year's events possible. Our platinum sponsors, the University Health Network, the Krembil Foundation, and TELUS Health. Our gold sponsors, listed again on the slide, the University of Toronto, London Drugs, McGill University, Moderna Canada, Women's College Hospital, Sunnybrook Research Institute, CAMH, McMaster University, and IDRC, and all our other sponsors and donors. Thank you again. And thank you for all the people who volunteered in the different institutes and universities across the country to help us bring this Science Week, not just here in Toronto, but across Canada. It's an important week of networking, engagement, and science and education. And I'm thrilled to welcome this year's 2023 Canada Gairdner Award Laureates in person, all of them, to join us today. As part of Gairdner's mission to inspire the next generation of researchers, our laureates, and some of our previous laureates as well, have been across the country sharing their research with universities, trainees, and high school students. It's always a special part of our program, and the inspiration of our laureates is really much appreciated by all. Today we're going to hear lectures from the 8 2023 Canada Gairdner Award Laureates about their research, and we're also going to hear short talks from 2 of our Canadian early career investigators who were selected by our laureates to present their related research and meet their scientific heroes. And now I am going to step away and hand over the podium to a series of chairs for the rest of today's events. And our first chair of the day is Brad Wouters, who is Executive Vice President, Science and Research, University Health Network. Brad.
Good morning, everyone. It's my pleasure to introduce our first laureates this morning. Our first speaker is Dr. Michael R. Silverman. The 2023 Canada Gairdner International Award will be presenting on Treasure Hunt, Searching for Lux Genes. Dr. Silverman was born in Fort Collins, Colorado. He was the son of a rural veterinarian who practiced in western Nebraska. And in high school, Mike studied vocational agriculture and later worked on an experimental farm where he developed an interest in plant diseases and bacteriology. He received his Bachelor of Science and Master's of Science degrees in bacteriology from the University of Nebraska. And in 1972, he completed his PhD at the University of California, San Diego, studying the molecular genetics of motility and chemotaxis in E. coli. This work required the application of classical and modern genetic methods such as DNA cloning and sequencing, gene product programming, and transposon mutagenesis. He continued research as an independent science at the Agoran Institute and as adjunct professor of marine biology at Scripps Institute of Oceanography in La Jolla, California. And it was there that he investigated motility and bioluminescence in marine bacteria, and his work with Joanne Engelbrecht and Bonnie Bassler resulted in the discovery of fundamental genetic mechanisms that control bioluminescence, and these mechanisms were later found to control many different functions in many species of bacteria. Mike retired in 2000 to the mountains of Wyoming and is back today for our first lecture. Please welcome Mike to the stage.
Well, thank you. Here I am, more than 20 years after I started working on this phenomenon, and there we go. Can you hear me? And well, I'm kind of stunned. What happened after all these years is all this excitement and commotion and prizes and articles written, and I'm just amazed what's become of this field. It's a big deal now, but when I started studying this, well, it didn't exist. This field didn't exist. There were a few clues around, and I like to use the idea of doing research as a treasure hunt. And there were a few clues, and then we started digging in and finding more clues and more questions and putting things together. And after years and years, that story really expanded. Anyway, I got into studying these bacteria that communicate. They communicate with each other. They communicate with other species. The communication results in many different kinds of responses. But the clue that got it going, can I have that first? Or I've got it, okay. The clues that existed when I started more than 20 years ago were in bioluminescent marine bacteria. And as you see in the 70s, Nielsen and Hastings discovered that the expression of bioluminescence was controlled by the density of the culture. If there weren't many of these bugs around, they actually didn't produce any light. But when they got together in dense concentrations, they started to glow, and they glowed a lot. They also showed, Nielsen and Hastings, that what they were detecting was something that they themselves made. That's why they called it an autoinducer, a chemical substance. And later on, Eberhard identified this particular autoinducer as a homoserine lactone. Now, this communication gets much more complex, and it's been found to exist in almost all bacteria, controlling all kinds of functions. But it's chemical signaling. But I like to think of bacteria as little people, real little people. And so I use the metaphor that they're talking to each other. So just that you know, they don't talk to each other. They, well, it's more like they smell each other. Anyway, the clues that existed more than 20 years ago were in these bioluminescent marine bacteria. And why was I so lucky to stumble upon the one clue around which would lead to this huge explosion of research? And the reason was I had this patron, the Office of Naval Research, which is part of the U.S. Navy, and they were interested in light in the ocean, natural light. And And also, I was just plain fascinated. Bacteriologists are kind of sensory deprived. I mean, you guys, your pet barks and bites and fetches, but my pet, well, I can't even see it. It's invisible. And when they glow in the dark, it's kind of a visceral attraction. It's like kids playing with fire. I think you would like it too. Anyway, about the time we started, we were excited, even evangelical about the power of genetics, because the recombinant DNA technology and the DNA sequencing technology had just been discovered, and we're really excited to put genetics to work. As you'll see, it's powerful in so many different ways. Anyway, we started with a marine bacterium called Vibrio fischeri, which inhabits the light organs of fish and squid. And the idea here was to try to clone a gene-sized piece of DNA that encoded this characteristic. What you do is you take Vibrio fischeri DNA and we'd cut it up with our molecular scissors into gene-sized pieces, and then we would link those to a plasmid vector, because you have to have some means of ensuring their replication. So you take the fragments, the little pieces of DNA, and you fuse them to another piece of DNA that will assure that they replicate. And then you take this DNA and you put it into another organism. We put it in E. coli. E. coli isn't luminescent, but it's the favorite pet research tool of bacteriologists. And if we're lucky, we'll find a recombinant that has the genes that encode the characteristic that we're looking for, which is bioluminescence. Well, it's kind of a long shot, because it presumes that all of the genes necessary for programming the production of light are on one piece of DNA. But it's easy. It's the easiest way to clone. There are many other techniques we tried. And, well, bingo. It worked. We got a recombinant, a piece of DNA, which when you put it into E. coli, would cause them to glow in the dark. Wow, that was really neat. We were really excited. That's E. coli glowing in the dark. E. coli has never did that before. We got really excited. We started making all kinds of bacteria glow. It was like Tinkerbell and Peter Pan, and we go around and make different things glow. Pretty exciting. Well, not only did this piece of DNA program bioluminescence in E. coli, but it worked the way it's supposed to work. This shows what the parent of this DNA, Vibrio fischeri, does. If you take a dense culture, it's glowing, and you dilute it, say, 1,000-fold into fresh medium, immediately the light starts going out. The light per cell goes down precipitously because this inducer substance is a very low concentration. And then as the culture grows and gets more dense, the light comes back on and it increases rapidly. And it could be 1,000, 10,000-fold more light coming out than when it was diluted. And so the recombinant looked just like the parent organism. So that was good news. We had the genes for light production, and the regulation was intact too, and we were interested in the regulation. How does that work? Well, the goodies were all on one piece of DNA, and it was arranged like you see there. Luxi, I like that name, Lux. It's just, it just tells you it's for light. And when you isolate a new gene, you get to name it. Ha ha. Okay, so I named it. Anyway, Luxi encodes a protein that makes this autoinducer substance. It's in a group of genes in a linear array that are read from left to right. At low cell density, there's not much of any of the expression of any of those genes in that large linear array, which we call an operon. Then over on the left, there's another gene read in the opposite direction that makes something called LuxR, and that's the sensor. So Luxi makes the signal, LuxR makes the sensor. Then, as this autoinducer accumulates, then it interacts with LuxR, and LuxR is an activator of transcription, a reading of this whole operon going to the right. So it's actually has a feedback, a positive feedback loop. So once things get going, it really takes off. Joanne Engelbrecht, she was a graduate student with me. I don't like to say she's my graduate student, but she was. And so, so anyway, this is the first of these sensing circuits to be discovered. And if you use this metaphor talking, Vibrio fischeri talks to itself. And it's one of the simplest circuits that we discovered. Now, before I go on to another organism, which Bonnie actually studied, and which was also much more complex. We weren't finished with the fischeri. We got to thinking, gee, this is easy research. All you have to do is look for light. Other types of scientists, they study other characteristics, and bear in mind the organism itself is invisible. Until there's a big pile of it, of course, and you get hundreds of millions of them together, then you can see that. But it was really appealing to be able to measure gene expression simply by looking at the light. And so we got to thinking, we can move this construct into all sorts of bacteria and make them grow. Wouldn't it be nice if we could use the same characteristic to measure with other kinds of functions, other kinds of properties? And so Joanne, she took it says luciferase and aldehyde there. That's actually 5 genes making and coding 5 proteins to make light. And Joanne wanted to find a way to use those genes as reporters. And what she wanted to do is take those luminescence genes and link them to other genes. We'll call them gene X, or the target gene. And I'm going to have a really hard time explaining this because the gadget that she built, it's so amazing, and it's just plain wacko. UC LuxCDABE. It's on a device that can hop or transpose. Believe it or not, there are DNA elements which levitate other DNA, other genes around. These things exist in us, they exist in bacteria. They're called transposons. And what Joanne did is she took LuxCDABE DNA and she flanked it with parts of these transposons, these gadgets that levitate genes. Pretty hard to believe, but they exist. And so Joanne built that. Actually, it started from a device that Malcolm Casadabon invented. Anyway, I'll show you how it works. You go after a target gene. The idea is to put this jumping gene setup, have it jump into gene X, then instead of measuring gene X's function, you just open your eyes because the light you see will be under the control of gene X. So there you see when gene X is off, the CDABE don't get red, and so it's dark. And then when gene X comes on, so does the light. And I'll show you how it actually works with an example in a non-luminous Vibrio called Vibrio parahemolyticus. It's sort of pathogenic, but not very. Anyway, it's a curious organism. It can get around. It's modal. See the little squiggles there? Those are flagella. And it can move around in a purposeful way. And so that's what it uses to swim. But when you grow it on a surface, or when it's out in nature growing in a biofilm, it changes. It gets very long and it gets covered with this fuzz, which is actually a different kind of flagella called lateral flagella. And that allows it to creep along over the surface. We call that swarming. So on liquid it swims, on solids it swarms. Now, we found that to be really puzzling. How do these bacteria know they're growing on the surface? Do they have a sense of touch? Well, how do they know? Anyway, what we did is we used Joanne's magic device to make fusions with the Lux genes and these swarmer genes. which we call LAF. And of course, this magical device of Joanne's, it also makes mutants when it's fusing the Lux genes to the other genes. And this is an example. You can see that on the right, there's a control. It glows whether it's on a plate or grown in liquid. On the left, you see the fusion of the Lux genes to the swarmer genes. And it only expresses those when you grow it on a plate, and you grow it in liquid, and the genes are off and the cells are dark. Well, now it's much easier to study this characteristic. I don't have time to tell you what we learned. No, I won't tell you. It's too long a story. But this bacterium does have a sense of touch. And if you catch me later on, I'll tell you how it knows it's on the surface. Anyway, I've got to get back to talking about Bonnie's contribution. Now, the problem with her bug, Vibrio harveyi, is that we couldn't find any mutants. And if a geneticist can't find mutants, he's screwed. And when Bonnie came to the lab, we gave her the hardest problem we had, which was try to understand what's going on with Vibrio harveyi. And Vibrio harveyi lives out with the general public. It doesn't live in a light organ. It lives in a more complex environment. And Bonnie studied this for almost 4 years, and it was a tough problem. And what she found is a much more complex arrangement for signaling. Signaling system 1, it turns out it uses a homoserine-like autoinducer. We didn't know what 2 was, but the point is that there was redundancy here. There were 2 signaling systems, maybe even, there are even more minor ones. And so what was happening was that when you knocked one out, light was still on. Or if you knocked the other one out, light was still on. No apparent defect. So how are you going to sort this out when there are multiple pathways? And well, we did find some really weird mutants that allowed us to eliminate one, and then we could study the other. Anyway, we got most of the genes. We didn't get the signaling system 2 signal. Bonnie went on to have a very stellar career studying this kind of signaling, and she figured out what the other autoinducer was. And well, I don't think I'm stealing her thunder if I tell you that this was a really interesting signaling molecule. If Fisherite talks to its own species, this other system is more like Esperanto, where you talk to all species of bacteria. It's the one they use to talk to foreigners. Anyway, foreigners, I don't know about that. Anyway, this is what really got us to thinking that this communication might be much more significant and varied than we thought before. And of course, over the intervening years, we found that talking is between like bacteria and dissimilar bacteria, and maybe even with higher organisms. And Bonnie will get back to this. This is just an example of how you can characterize sensory system mutants. On the left, you see on the plate is a crescent. Both of those bacterial smears, both of those strains are dark. But the one on the left makes a substance that can stimulate the one on the right. You see that? After a few hours, something diffuses through the agar plate and stimulates those cells to glow. And if you wait longer, it diffuses farther. And there, right before your eyes, you're seeing chemical communication. The one on the right is dark because it's deficient in the signaling substance. So that's how we characterize. That's just a simple example of how we characterize these mutants. Now, we had a lot of fun doing this. Bonnie and I, and Linda and I, and Joanne and I, we worked for years together, and it's very gratifying to do science and work together. But what also happens that's very gratifying is you become friends. You become family, and that's one of the big benefits of doing research. Bonnie comes and visits me in my hideout in the mountains, and we talk about science, but we also go canoeing and hiking, and it's a great source of happiness to have friends like Bonnie. So thank you for your time.
That's terrific. Our next speaker this morning is Dr. Peter E. Greenberg. He's the 2023 Canada Gairdner International Award Laureate, and he will present on quorum sensing, a story of communication and cooperation. Dr. Greenberg was born in Hempstead, New York, received his bachelor's degree in biology from Western Washington University in 1970, and his PhD in microbiology in 1977 from the University of Massachusetts. After finishing postdoctoral studies at Harvard, he joined the faculty at Cornell University, and he later moved to the University of Iowa as professor of microbiology and Shepherd Professor of Microbiology, and finally joined the University of Washington in 2005, where he is currently the Nestor Professor of Microbiology. Dr. Greenberg has studied quorum sensing since the late 1970s and is widely considered the father of the field of microbial quorum sensing. The term quorum sensing originates from a 1994 Journal of Bacteriology article on which he was the senior author. Dr. Greenberg is a fellow of the American Academy of Arts and Sciences and a fellow of the National Academy of Sciences. Please join me in welcoming Dr. Greenberg to the stage.
The laser pointer has disappeared. Do we? I can use my finger. Yeah. Oh, down here. First, what an honor it is to be standing here telling you about what we've done for the last, I don't know how many years. It's not on? Tell me when it's good, Janet. So I just have to thank the Gairdner Foundation for recognizing the work that we've been doing. And I want to tell you about quorum sensing and what I call the new science of sociomicrobiology. What has sprung from all of this stuff Mike just told you about? First, why do we work on sociomicrobiology? Well, when I began, microbiologists didn't think bacteria were social. That was in the domain of higher organisms, but we've come to understand that they are, and that they're powerful tools for understanding the basic biology of sociality. And I'll give you some examples of that towards the end of the talk. You can do extraordinary things with bacteria. You can study social evolution on your benchtop through many, many generations over a couple of days. You can't do that with higher organisms. So I love bacteria. Just like Mike, there are pets, and if you want to understand bacteria, it's now clear that you can't ignore their social activities. They work as collectives, and if you want to understand how they do that, you have to think about sociality. And the Holy Grail is this idea that we can develop new medicines to treat bacterial infections. Well, we'll see if that's true. We all hope so. So I want to start just by telling you sort of my view of the stages of scientific discovery. So our colleagues first say what we're studying is not true. Then they say it's true, but it's not important. And finally they say, okay, it's true and important, but it's not new. So I want to sort of take you through the stages, starting when Mike and Bonnie and I were like the only people interested in this were the three of us for a while. And that was enough for us. We thought this was really cool, and we thought it was important, and at least we had a couple other people to talk to. So how did I get started? Well, I went to the famous marine biological laboratories in Woods Hole, Massachusetts one summer, the summer of 1973, to take a course on marine microbial ecology. And I met these two characters, Anatol Eberhard and Ken Nielsen, and they told me about these extraordinary luminescent bacteria, these bacteria that made light. And they had two species that Mike already introduced to you, Vibrio fischeri and Vibrio harveyi, that they said could auto-induce light, that they produced a chemical that somehow magically, when it accumulated in their environment, told all the bacteria to make light. So most microbiologists of the time thought this was nutty stuff, that bacteria were talking to each other. So people didn't take them seriously. I thought it was fascinating. And so in the summer in Woods Hole, the town is just full of biologists. And so a big night out, a big Friday night out is going to the Friday night lecture. That's what you do. And I went to, I did that. And they were interesting, and I was particularly fascinated by this guy, Ed Wilson. He's an ant biologist. He studies the social ants. And he was explaining how you could study sociality in any organism as a biologist. It's a point I'll come back to later. And I thought, wow, these bacteria that only make light when they're in a crowd are communicating to coordinate cooperative behavior. Nothing can see the light made by a few cells in a milliliter of whatever, but we can see the light of the collective. So I decided, well, maybe it's crazy, but I want to work on this. Well, I had to finish graduate school. I was working on something else. And then I wrote to Woody Hastings, who was the mentor of these guys at the time, and asked him if I could do a postdoc. He said, sure. He's a biochemist, so he took this microbiologist into the lab, and I like to say that he sort of viewed microbiologists as pets. We weren't serious scientists like biochemists. So I published, this is the first paper I published on what we now call quorum sensing, and I published with Woody Hastings, induction of luciferase synthesis by Beneckia harveyi by other marine bacteria. So this paper was a great disappointment to me. I wanted a communication system among kin. I didn't want bacteria that were listening to all sorts of things. And I have to say that I missed the big picture here. And you're going to hear about this later from Bonnie. She figured all of this out, and you'll find out how important it is. So I was busy, though, even though I missed this, I was busy. So you heard about Vibrio fischeri. So I talk about the Vibrio fischeri paradigm, communication and cooperation in bacteria. And when I started my lab at Cornell, I turned my attention to this species because induction of light required something this species produced, but it wasn't produced by other species. So this was a species-specific signal. This seemed more like communication to me. And you heard why they do this. So they live in the sea at very low population density, where it would be a waste of energy for them to make light. So they're at low density and they don't make light. They also live in light organs of certain marine animals. This is one of them that's been developed by these two scientists into a good laboratory model to study how luminous bacteria colonize light organs of marine fishes and squids. There's a light organ in here surrounding the ink sac, stuffed with a pure culture of Vibrio fischeri. So the squid give the Vibrio a good place to live, and the Vibrio give the squid light for its purposes. So it's a mutualistic symbiosis. So that explains why they want to control light production in a density-dependent fashion. So here we are in the 80s, all through the 80s, the three of us having a great time, and the rest of the biological community could care less. And Mike told you how this worked. So he told you that there was an autoinducer. I'm showing the structure of the autoinducer here. That was determined by Eberhard in 1981. It's a non-essential amino acid stitched to a fatty acid. And then Mike got these genes along with Joanne Ingebrecht, and that was an enormous breakthrough. That enabled us to really start moving fast. And so this was 81, 83, and then we showed how this works as a proxy for cell density. So cells make it inside, and the cell membrane is permeable to this molecule. So it diffuses in and out by passive diffusion. And at low population density, then it will always be diffusing out into the external world. When the cells are crammed together, it can start to accumulate, sort of matching concentration inside and outside, and it will bind to LuxR, as Mike said. We've learned a lot about how this works now, and LuxR will activate transcription of the luminescence genes. So now it's true, but it's not important. So we're still having fun. So this is a real example of where basic curiosity-driven, fundamental research can go. So fast forward a decade, and in the 1990s, we began to discover that it's not just Vibrio fischeri. So I want to talk about the birth of quorum sensing. In 1992, a group in Nottingham reported that other bacteria made this acylhomoserine lactone that Vibrio fischeri makes. And one of them actually controlled the production of antibiotic biosynthesis using this signal. So that was good. About the same time, a group in Australia discovered that this plant pathogen regulated its ability to transfer DNA from one cell to another by an acylhomoserine lactone. So there's no sense in being able to transfer your DNA to another cell if there's no other cells around. And then we discovered that this important human pathogen, Pseudomonas aeruginosa, controls a battery of virulence genes by two acyl HSLs that were similar to but not identical to the Vibrio fischeri system. So now, for microbiologists, it's true and it's important because a pathogen's doing this. You know, I sort of consider this a short-sighted view of what's important, but that's what my colleagues think. So at that time, we wrote a little mini review. We were starting to see a picture, and we wrote a little mini review on this in 1994, trying to think of a catchy title for what we were trying to capture. Quorum sensing in bacteria, the Luxar-Luxi family of cell density responsive transcription regulators. So that's where the term comes from. And that caught the attention of a lot of microbiologists for some reason that hit people's imagination. So now it's true and important. And then I had the good fortune that a really good science writer became interested in this. Her name is Evelyn Strauss, and she wrote an article for a general science magazine in the 1990s, and she was trying to capture what we were doing, and she called this mob action, peer pressure in the bacterial world, that the group could force the individuals to do something. She talked about bacterial barn raising, and this is an important concept, that these little creatures can do so much, and they do it because they can organize and cooperate and work as a collective. So what do we know? Quorum sensing is a kind of bacterial cell-cell signaling. There are now lots of other mechanisms besides this acylhomoserine lactone mechanism. This is the one that we've concentrated on. It's a kind of cell-cell signaling used to control specific genes, and it allows coordination of group activities. If there are activities of a group, that implies some sort of social interaction. And Bonnie might disagree with me about this. I like to say that the system that's most well studied is the acyl HSL quorum sensing system, and it exists in proteobacteria, lots of different proteobacteria, gram-negative bacteria. We now know of over 200 different species of bacteria that produce and respond to these signals, and they regulate different things in different species. Give you two examples now. The 7 Lux genes in Vibrio fischeri. Vibrio fischeri doesn't regulate much else besides these genes by quorum sensing. We found that Pseudomonas aeruginosa activates about 300 genes by quorum sensing. And those genes are spread all over the chromosome. So just a little bit about how Pseudomonas works. This was the big thing that made it true and important. So Pseudomonas has 2 Luxar-Luxi pairs. So the way we were able to discover these was in the 90s, DNA sequencing was becoming easier, and lots of microbiology labs were sequencing and identifying genes that coded for proteins that look kind of like Luxar. And this is one of them. It's called LASR, and it codes for a transcription factor that responds to so this is LASI, and the LASI product produces a C12 HSL. It's a longer side chain than the Vibrio fischeri AHL. And LASR, in the presence of sufficient C12 HSL, activates a constellation of genes. We call it the LASR regulon, including, at least in our laboratory strain, this second set of Luxar-Luxi homologs, RELR and RELI. And RELI codes for an enzyme that makes a C4 HSL. That's shown here. And RELR responds to that and activates an overlapping set of genes. So it's controlling virulence genes. And it turns out that a lot of the genes that are activated code for the production of extracellular things. So a little bit about the nuts and bolts of how these things work. So then it's important, and we had a lot of fun finding out details about how Luxar homologs and Luxi homologs work. So Luxi homologs, well, the enzymology is reasonably well studied. The HSL comes from a common cellular metabolite, acetylmethionine. The acid side chain, the acid part has to be activated. So it's a high energy intermediate, and it comes in 2 flavors. It can be activated by acyl carrier protein from fatty acid biosynthesis pathways, or it can be activated by coenzyme A from acid degradation pathways, or from general metabolic pathways. So there are 2 different flavors, and we can now predict from DNA sequence which will be an ACP type or which will be a CoA type. So this shows structures, solved structures of several of these I proteins from different bacteria, some of which are CoA type and some of which are ACP type. And you can see the only thing I want to say is that these structures are very similar. What we can't do yet, which I really want to be able to do, is predict from sequence what will be the signal produced by one of these. So there are now almost 4,000 Luxar-Luxi pairs in the DNA sequence databases that have accumulated over the years. And in order for us to figure out what a Luxi is making, we have to do chemistry. I want to be able to skip that step. I want my AI friends to help us out here. So, I mean, there's lots of questions about these Luxi proteins. The Luxar types, by now several Luxar homolog crystal structures are available, and of course with our AI friends, it's now possible to reliably predict the structures from sequences, and we know that these function as dimers, and each monomer in the dimer consists of 2 domains, an N-terminal domain that binds the AHL, and a C-terminal domain that binds to target DNA and then allows activation of genes in most cases. And we're beginning to learn by computational tricks about how these pairs, so they're always cognate pairs, Luxar homolog and a Luxi homolog go together. We want to know how all the diversity arose. I think I skipped this little part of the last slide. This gives you a smattering of the types of structures that we found. We've now found over 30 different types of AHLs produced by different bacteria. This all presumably came from a common ancestor. How did that occur? So we've had trouble changing specificity, evolving specificity in the laboratory, but we can do it by identifying co-evolved amino acids and changing them, and thus changing what one of these will make and what one of these will respond to. And we're hoping to be able to learn about the evolution. So first we figured out how. We figured out the nuts and bolts, and we figured out about signals in other bacteria. And then, the big question is why? That's the harder question, right? And we sort of had in plain sight why for Vibrio fischeri. So it doesn't want to make light in seawater. It wants to make light when it's in a symbiosis. And its light can be seen as a collective activity. Light has no purpose for a loner. But what about other bacteria like Pseudomonas? Does this hold up, or is it just an analogy? So we had learned what sort of genes were controlled by Pseudomonas, by quorum sensing and Pseudomonas. And among the genes are genes that encode extracellular proteases, enzymes that the cells secrete, and that enables Pseudomonas to grow on protein. So these little creatures can't transport in big proteins. They have to break them up outside and then transport them in. So these proteases are like public goods, common goods. They're shared amongst the population. So here's the idea. Pseudomonas makes proteases, secretes them. The proteases break down protein in the environment and into this yellow stuff, which can diffuse around. This is Pseudomonas food. And so we call the proteases public goods. And here I've labeled these blue cells as cooperators. So they're all contributing to the public good. And evolutionary theory says that there should be some selection for what I'll call a cheat, the brown cell here that doesn't make the protease itself, but it accrues the benefit of the protease made by the collective. So it has a fitness advantage. And cheats should take over, right? So the first test of the hypothesis that Pseudomonas can control cooperation by quorum sensing is to culture it in a medium that only has protein for it to grow on. And cheats should emerge. And so you can do that kind of experiment. It's shown here. At the beginning of the experiment, there aren't enough cheats to detect. But transferring daily, after about 2 weeks, you see them start to emerge. And they reach about 40% of the population. So somehow the cooperators can control them. They don't take over. And we can begin to understand at a molecular genetic level how cooperators can control cheats. And this is something that really excites me. So these are protease negatives that come up. And by sequencing, you can show that all the protease negatives are LASR mutants. So they're defective in the quorum sensing activator at the top of the quorum sensing cascade that is required for production of proteases. So let's see where we are. We're still working on various nuts and bolts of these systems, and we're still working on why bacteria do this. And a big question is, under what real life conditions are they using these systems? So we've been studying quorum sensing in test tubes almost exclusively. And we have a lot to learn about how this is implemented in the real world. We have been studying how cooperators control cheats. And we can make mutants that can't control cheats, for example. And then that's enabling us to understand what the basis of cheater control is. So this provides fundamental information about the biology of cooperation, right? So we're learning about the genetics of cooperation. And it's essential as the field aims to target quorum sensing with therapeutics to control infections. We need to learn about all this if people making quorum sensing inhibitors have any hope of deploying them in the right way and getting them to do something useful. So I think that is all of science. I just want to give you a little bit of my crystal ball now that it's true and it's important. My wish list of things that we want to learn. We want to be able to predict signal chemistry based on sequence information of R or I polypeptides. We'd like to be able to evolve diverse signaling systems in the laboratory. We'd like to understand why many bacteria have multiple AHL gene pairs. So I told you Pseudomonas has 2. Some bacteria have as many as 7. Why do they need so many? This is a big area, AHL quorum sensing in the context of polymicrobial communities. Can you imagine all the noise in a mixed microbial community? How is that sorted? And we're learning about the principles of communication and cooperation at a molecular genetic level. And this is the Holy Grail, of course. The idea is to develop new medicines, and my little contribution is trying to learn more about what we need for others to succeed. I don't know how to make medicines myself. Just to finish, a little on some more armchair philosophy. It's now not new, but it's important, and we need to maintain a field I call a field with legs. The field is matured, but I hope it's aging well and that it has a long, I'm sure it has a long, exciting run ahead. The point is, in science, it's important to encourage an interactive collegial discipline as it grows, to bring new people in, new ideas, and welcome them, and it keeps the field alive. And I think that we've done that as best we can. And I will stop there, and thank you for your attention.
Well, terrific. So my next introduction is for someone we've heard a little bit about already, Dr. Bonnie Bassler. She is also a 2023 Canada Gairdner International Award Laureate, and she will present on quorum sensing across domains from viruses to bacteria to eukaryotes. Bonnie Bassler is a Howard Hughes Medical Institute investigator and the Squibb Professor and Chair of the Department of Molecular Biology at Princeton University. She grew up in Northern California, and as a young person, she adored nature and animals and hoped to be a veterinarian when she grew up. However, she became fascinated with biochemistry and molecular biology when she went to college and switched directions. She received her Bachelor of Science in Biochemistry from the University of California at Davis and a PhD in Biochemistry from Johns Hopkins University. As you heard, she performed postdoctoral work with Michael Silverman and joined the Princeton faculty in 1994, and her research focuses on molecular mechanisms that bacteria use for intercellular communication or quorum sensing. Bonnie's discoveries are paving the way to novel therapies to combat disease-causing bacteria. She has received numerous prizes, including the MacArthur Foundation Fellowship, the Shaw Prize in Life Sciences and Medicine, the Dixon Prize in Medicine, the Gruber Genetics Prize, and the Wolf Prize in Chemistry. Bonnie also received the Princeton President's Award for Distinguished Teaching. She is devoted to diversity in the sciences and educating lay people about the thrill and relevance of research. She was also president of the American Society of Microbiology and served on the National Science Board, to which she was nominated by the US President Barack Obama. The board oversees the National Science Foundation and prioritizes the United States research and educational activities in science, math, and engineering. Please join me in welcoming Dr. Bassler to the stage.
Well, thank you. I'm so delighted to be here, and it's such an honor to win this prize with my colleagues. And so I want to thank the Gairdner Foundation for highlighting us, our teams, and our field. And what I thought I would do, since Mike gave you a historic talk and Pete gave you kind of a middle, half historic, half new, is try to show you our field is flourishing and tell you about a brand new project in my lab. So that's where we're going. Okay, so you guys already know that I study how bacteria get any bang for their buck. You know, so they're these itty bitty critters, they're puny, yet they have all this power. They can kill us, and they can also keep us alive with our microbiome. So what we've always wondered is how do bacteria manage to do those fantastic feats? And I'll give you one slide that you already know about. They do it by chemical communication. So they communicate, they count, they recognize when they have the right number of cells, that if they all do something together, they can accomplish feats that they could never accomplish if they acted as individuals, because individually, they're too small to make a difference. And so now you know we call this process quorum sensing. And so please forgive that you're going to get one more, you get my version of it. So what we know now is bacteria have to distinguish times when they're alone from times when they're in communities, so they can act appropriately under those two scenarios. And so the way they do it is by making and releasing these small molecules that Mike and Pete told you are called autoinducers. The world is big, bacteria are small. When bacteria are alone, the autoinducers diffuse away, they can't detect them, and that says act as an individual. But as the cells divide, since all of the bacteria are making a share of the autoinducer, the extracellular concentration of the chemical increases in proportion to cell number. And when the molecule hits a particular threshold amount, the bacteria detect it, and they infer that they must have neighbors around. And so in unison, they all change their behaviors together, and they begin to carry out collective or group behaviors that takes lots of bacteria doing them simultaneously for the behaviors to become effective. And so, of course, I've shown the bacteria in blue because this all came from these bioluminescent bacteria. You heard that from Mike. And then what you heard from Pete is that we now know that quorum sensing is the norm in the bacterial world. And in every case that's been, so there are thousands of cases of quorum sensing known, and in every case that's been studied, hundreds of genes are controlled by these quorum sensing circuits. So it's a big deal when a bacterium decides to change from going it alone to acting as part of a group. And then we know a lot about these molecules, and both Pete and Mike alluded to my part of this game. What we now understand is it takes multiple molecules to have a proper quorum sensing conversation. So at least what I'll show you 4 molecules. So let's start with 3. So first, there's always, so the molecules I'm showing you are from my favorite Vibrio, but the principles that I'm going to tell you about transcend all quorum sensing systems. So I'm just going to show you the particular molecules, but you can use your imagination. The bacteria tinker with the structures, they tinker with how they detect them, but the ideas I'm telling you are generic. So there's always a molecule, one of these homoserine lactone molecules, that as far as we can tell, one and only one species of bacteria make. So this molecule is for intra-species communication. This says, I am your twin. I am your clone. Then there's a molecule we discovered, that this fatty acid, that all Vibrios make, but as far as we can tell, nobody but Vibrios makes. So this is the molecule for the genus. So the first molecule says, I'm your twin. The second molecule says, I'm your cousin. Then the third molecule we discovered is the universal molecule that Mike told you about. All bacteria make exactly the same molecule, so there's no species information in this molecule. It's the molecule of other. So what we now know is that these molecules encode something about cell number, but also something about how closely or distantly I am related to my neighbor. And so the sort of miraculous computation that we now know bacteria do is they actually measure the ratio of these molecules. So what I think the bacteria are doing, the first thing I think they do is they ask, am I alone? Or am I in a group? And so they begin to detect these quorum-sensing molecules, and that starts to set this program of gene expression at the bottom of the cascade. But then the more sophisticated computation that they do is they measure the ratios of the molecules. So what we think they're doing is they're asking, is it me and my kin that are in the majority, or is it the enemy? And then they tailor the program of gene expression they turn on based on whether they and their family members are in the majority or whether their foes are in the majority. So what we know now is that they can tell self from other and friend from foe using these molecules, and they act appropriately based on the number of bacteria that are around them and who those bacteria are. Okay, so that is my view of quorum sensing. So now what I want to do is move to this new project that we're working on, and I'm going to tell you about a story in a pathogen called Vibrio cholera. So you probably know that Vibrio cholera is endemic in underdeveloped regions of the world, and it causes this terrible diarrheal disease that people get when they drink contaminated water, eat contaminated food. So my gang showed that cholera has quorum sensing, it has the molecules I showed you, and that quorum sensing is essential for cholera to be a pathogen, because quorum sensing controls transitions between infection and dissemination. But cholera is a really persnickety bacteria that has this insidious program. What it does is when it gets into the human host at low cell density, it comes in guns loaded, it's spewing out its toxins, it's making a biofilm. So at low cell density, when it infects the host, it's highly virulent, it's making a biofilm, and that's how it gets in. But then once inside the human intestine, it begins to grow and divide. Quorum sensing autoinducers kick in, and at high cell density, what the autoinducers do is it tells cholera to stop being a pathogen, to turn on an escape program, and out cholera comes by the gazillions to infect the next patient, right? So it's all about dissemination. So quorum sensing controls these transitions. Cholera has to have quorum sensing to do this, but quorum sensing shuts down virulence and turns on an escape program. Okay, so we had discovered that a few years ago. Several years ago, and then a couple of years ago, a fantastic rotation student named Justin Silpe came to the lab, and he was studying this system. And he discovered the fourth quorum sensing system in cholera. So what Justin found was a transcription factor, a cytoplasmic transcription factor. It's a quorum sensing receptor that he named VQMA, for Vibrio quorum A. And so just like I told you, at low cell density, that turns on virulence, turns on biofilm formation. Then at high cell density, when the autoinducers accumulate, the autoinducer binds VQMA directly, and that shuts off virulence and turns on this escape program. So it was a brand new quorum sensing system in cholera, but the logic is identical to what I just showed you about the canonical quorum sensing systems in cholera that we had already discovered. It's all about getting out. And then what Justin did is he went on to discover what this new autoinducer that we call DPO is. And so this is the structure that he determined. DPO is like all of these quorum sensing molecules, is a simple molecule. This molecule is actually made from threonine. This half of the molecule is made from threonine, and this half of the molecule is made from alanine. So 2 amino acids, and I'll come back to that at the beginning. So it's a really simple autoinducer, but it's a brand new molecule to mankind. And I should say that about all of these molecules that we've discovered, they're all really simple. They're made from ancient substrates, but every time they're brand new molecules. So these bacteria are just treasure troves of these new chemicals. All right, so you get it at high cell density, this system kicks in and turns off virulence and turns on the escape program. So once Justin had gotten this far, he was a rotation student. I hope I sound really nice, but I wasn't gonna let him graduate, and because he was a rotation student, so he had to like figure out his next project. And so of course he was enamored with his own discovery and the system that he had found. And so he was kind of farming around trying to figure out what to do next. And so he was looking at databases, trying to understand the role of this new system. And he found this interesting thing, which was that this protein, VQMA, was in every Vibrio. So every sequenced Vibrio has a VQMA protein, but no bacterium except a Vibrio has VQMA. So it's a Vibrio gene. By contrast, DPO is broadly made in the bacterial world. We have tons of bacteria that make this autoinducer. So there seemed to be this asymmetry between perhaps reception of the molecule and production of the molecule. And he was trying to figure out what that pattern meant, and we haven't figured that out yet. But he did actually find something that got us sidetracked to work on when he was thinking about this. He found one more VQMA, but it wasn't in a bacterium. He found a VQMA gene on a phage, on a virus. So phages are viruses that infect bacteria. So there was a virus genome sequence in a database. Nobody had studied this virus. We got it in the lab, and what was interesting is that it looked like it had a quorum sensing receptor gene on this viral genome. And that was weird because quorum sensing is supposed to be about bacteria. So we got and this phage is a Vibrio phage. It infects Vibrios. But what's interesting about this phage is it lives as a linear plasmid. So it never integrates into the chromosome. It just lives in its host cells as a plasmid. And so we sequenced the phage, and I have things color-coded. And on the right-hand side of this genome, there were genes that we could understand from classic seminal work that was done on, say, phage lambda 70 years ago. So phages like this one and like lambda, phages that you've heard about, when they get temperate phages, when they get into a host bacterial cell, they have to decide stay or go. So that's called lysogeny, where they just get passed down through generations, or they can replicate like crazy, kill the host cell, and leave. So it's lysogeny or lysis, lysogeny or lysis. Phages have to decide that upon infection, and these are the genes that mediate that decision in all these classic studies and in ours. So what Justin did were some of these experiments that had been very similar to what was done in lambda. So what happens is a phage will merrily live along in its host until the host cell gets in trouble. So whenever there's DNA damage or SOS and the host is stressed out, the phage says, that's it, I'm out of here. And what happens is that this protein C1 gets cleaved. And that inactivates it, it's a repressor. And so that allows this component Q to be made, and Q turns on all of the lysis genes. So C1 gets cleaved, Q launches, the phage replicates and kills the host and moves on to the next victim. So we could see those genes on our phage. Justin did those. We did not need to rediscover that. That had already been discovered 70 years ago. This works like a textbook in our phage. Our question was over on the left-hand side of the genome. Why is there a quorum sensing receptor on a phage, and why is our quorum sensing receptor on this phage? And so that's what we really wanted to study. And so I had told you that Justin had just discovered DPO. And so we only knew how to do one thing, which is to add DPO, the autoinducer, and see what happens. So we had a synthetic procedure to make DPO, and we did the experiment. So then what Justin did was he infected Vibrio cholera. Okay, so now cholera is carrying the phage. And then he just did a growth curve. And so what you can see is that cholera grows fine, even though the phage is present. But if Justin adds DPO, the autoinducer, to the infected cholera, what happens is that the phage receptor detects it, and now look at the white circles, and all the cholera die. And so our interpretation of that result is that these phages are eavesdropping on host quorum sensing. They're surveilling the autoinducer, they're monitoring host cell growth, and they're killing the host exclusively at high cell density. And so if you think about it from the phage's point of view, if a phage is going to pop out of its current host, if there's no other host to get into, it's a goner. So when's a good time for phage to kill its host? Well, it's when it's at high cell density. And so apparently the phage is eavesdropping on the host in order to understand the perfect quote unquote, density to kill the host. And so for all of the 70 years that we've known about phages, it's always been thought that the only thing phages can tune into is host SOS or DNA damage. And so Justin, at least with this example, would say that can't be true, that in fact, at least this phage can listen in to host sensory information and input that information into its lifestyle decision making. All right, so it's eavesdropping. So then the next question for Justin is, how is that happening? How is this phage quorum sensing system connected to this textbook lysis-lysogeny machinery that we knew was there? And so you're going to remember this is a transcription factor. It binds DNA when it's bound to the autoinducer. So we thought this is really simple. What's going to happen is that the autoinducer bound to the receptor at high cell density will turn on Q, and then lysis will happen. So Justin tested that, completely wrong. We said, okay, it could be a repressor of a repressor. That would also work. So Justin tested whether it worked on C1, totally wrong. And so that left us with no genes that we understood. And so we had to infer that there must be some new component that completes this circuit and somehow links phage quorum sensing to the lysis machinery. And so Justin wanted to find that. So he did a genetic screen. He just chopped up the phage genome and looked for genes that would complete the circuit to lysis. And sure enough, he found that. It turned out there was one gene required, and it was this tiny little gene that I have in red that was sitting right next door to the quorum sensing receptor, but running in the opposite direction. This gene encodes a 78 amino acid protein, so a tiny, tiny little protein. And what was interesting when Justin took this gene and put it in all databases is that there were no homologs. So it was a brand new protein, brand new gene. So as you heard, Justin got to name it. You get to name it if it's your own new gene. So he named this gene and protein Q-tip, because Justin does not care about my reputation. So he tells me that this rolls off your tongue, that Q-tip stands for quorum-triggered inactivator of C1 protein. Q-tip. Okay, so the question then is, what does Q-tip do to connect these two phage lifestyle programs? And so Justin did a trillion experiments, and obviously I'm just going to show you the one that worked. So what Q-tip does is it sequesters that C1 repressor of lysis. So what you're looking at here, this is E. coli. You're just looking at them under a microscope. And what Justin's done is he's cloned into E. coli's C1, but he put a halo tag on it so they turn green. And so what you can see is all of the C1 protein is diffuse in the cytoplasm of the E. coli. If Justin also clones into them Q-tip, now what you can see is that all of the Q-tip makes C1 go into these big oligomers, and it sort of drops out as inclusion bodies at the poles of the cells. And so it sequesters C1 and inactivates it. And they look like Q-tips. It's as funny as it gets in my lab. Anyway, all right, so that's how we got that protein and gave it its name. And so then getting back to trying to pretend I'm a serious scientist, this C1 is active, this C1 is inactive. Okay, so the job is to make this big glop, and C1 is inactive. So then what happens then is that C1 has two fates. SOS, it gets cleaved and inactivated. Quorum sensing, it gets sequestered and inactivated. So the fate of C1 is different, but the outcome to the cells is the same. They are killed, and the phage moves on. Okay, so once Justin figured out the beginnings of this mechanism and we had this phage, we worried that this was just some accident of evolution, that this phage had picked up this VQMA gene, and so we worried it was a one-off. But now that Justin had this module with both of these genes, he could do much more sophisticated database analyses, and it turns out this isn't a one-off. We have now found hundreds and hundreds of phages, none of which have been studied, all of which have these two gene modules in that they have a transcription factor that detects some input, and then next door they have these novel anti-repressors that work by the same mechanism, which is to sequester their C1s and then allow phages to detect host information and connect it to these lifestyle decisions. So it's not a one-off. We've found tons and tons of these. By now, we've even found phages that have receptors and autoinducer genes. And so we think that this is going to be a very, hopefully, common way of these phages detecting extrasensory input. So then what I'm telling you, right, is that in our phage that hadn't been studied, the receptor detects the autoinducer DPO at high cell density and then launches this pathway. All of these other phages, they're not all quorum sensing transcription factors. We don't know what these phages are listening into. We presume that in their particular hosts, in their particular niches, these receptors detect some appropriately environmental signal, and then they connect that input to this new anti-repressor type mechanism to control lifestyle decisions. And so we're really keen to be studying these new autoinduced, or these new sensory systems that exist in phages. So once we got there, what Justin thought, Justin was trained as an engineer initially, and so what he thought is what these phages are doing is they're rewiring these circuits and connecting them to the lysis decision. And he thought, well, heck, if the phages can do that, I can do that. And so what we wanted to do is to take our basic knowledge and then try to make something practical, some applications that perhaps would be good for our colleagues that do more applied biology. And what I mean by that is we wanted to make phage therapies. So Justin made about 10 of these sort of toy phage therapies. I'm just going to show you one for an example, and then you can use your imagination. But the idea is we could engineer phages to kill on demand. And what you're also going to remember, I told you that's crucial about this family of phages, is they're linear phages. So it's not like they have host specificity. You can just transform these phages into any bacterium as far as we know. So for example, let me just show you the one example. So here's our phage. You know all the genes and the colors. And what you know is it's all about Q. When Q turns on, the phage is going to kill its host. So what Justin did, as one example, was he took a promoter from Salmonella, right? So Salmonella doesn't have anything to do with Vibrios, right? Salmonella is Salmonella. It causes this diarrheal disease, food poisoning. And the way that that works is that Salmonella has a promoter for genes called invasion. So this INV-F promoter in Salmonella turns on invasion genes when Salmonella is a pathogen. So what Justin did was he took these 100 bases of DNA and he just engineered them onto the phage. So we have this recombinant phage. So the way it works in Salmonella is when Salmonella is in virulence conditions, a transcription factor called HIL-A detects virulence conditions, it engages, and it binds at INV-F and turns on invasion genes. However, if this recombinant phage is in Salmonella and then we put it under virulence conditions, HIL-A does its job. It goes and it binds in INV-F, but that's on the phage, so all the phages replicate and all the Salmonella commit suicide, right? So you get it, we can get these phages to kill in response to inputs that we decide. And so that's the Salmonella one. You know, you get it. You can put any target that you can control, IPTG, arabinose, you know, whatever you want. You can actually use these little phages to kill when you want. And they don't transfer to other bacteria because they're living as plasmids. So anyway, we're hoping that this kind of technology could be useful for colleagues of ours, and you guys get the idea. All right, so let me finish up and go back to the basic studies and try to put together what I've told you in this Gairdner talk. So when cholera gets into the human host, it has to decide stay or go, stay or go. And that decision is mediated by quorum sensing. In this case, by this new quorum sensing system that has this DPO autoinducer that binds to the VQMA receptor, and then cholera disperses at high cell density. You're going to remember I told you that lots of bacteria make DPO. Well, that includes your microbiome. So it turns out in a mouse, maybe in a human, that depending on what microbiome is there and how much DPO they make, what happens is they're also contributing DPO to this situation, and they make cholera miscount. It counts it too high, too early, and what happens is that the mouse gets less severe cholera disease. It's known that humans can get more or less severe cholera. We have a fantasy that that has to do with the blend of the microbiome and perhaps how much DPO is being made. So what's obviously clear, though, you know, that may or may not be true about severity in humans, but what is clear is that the pathogen is trying to count with DPO, and your microbiome is fending off this pathogen using this molecule. Your microbiome lives lives in harmony with your intestinal cells because one of the things your intestinal cells supply is a protein called mucin. So mucin covers our intestinal cells. It keeps everything nice and gooey. It keeps everything in harmony. And microbiome bacteria have mucinases. So they eat mucin for a living. They're supposed to. But it turns out that every third amino acid in mucin is threonine. And you're going to remember I told you that DPO is made from alanine and threonine. So this is a three-way conversation in that you, the human host, supply your microbiome the threonine it needs to make this autoinducer to fend off this pathogen. So the eukaryotes, the bacteria, and the pathogen are all involved. And then in today's story, there's the parasite of the parasite that's involved. So now we also have to get these viruses in this conversation, and they're also trying to optimize off this DPO molecule to decide the same thing, stay or go, in this case, in their bacterial host. And so I'll finish my little talk by saying we don't understand how this works. We don't understand anymore how anyone counts correctly, how you know who made it and how much is there and when it's the right thing to do these lifestyle changes. And so the horizon for my field, and Pete actually alluded to this, is to get out of these single cell in a flash shaking around experiments and begin to build experiments that look like the authentic situations that bacteria live in so that we can really start to understand how can quorum sensing possibly work when the complexity is changing and it's so high. And so that's what my gang is trying to do. We're trying to make our experiments look more like this and hopefully figure out new quorum sensing systems and actually figure out how quorum sensing works for each of these participants. And so I want to thank you again. Let me show you today's hero. This is Justin Silpe. I did not let him graduate, he graduated, right? So that was Justin Silpe. He did all of the work that I told you about the VQMA and the phage. He's just a spectacular scientist and lucky microbiology to have him. A couple years ago was my 25th anniversary at Princeton, so not too long before this. And so this is everybody who's worked in my lab and their spouses and their families. They all came back because I want to say that I'm telling you this one news story, but so many wonderful students and postdocs have come through the lab, and I'm standing here today absolutely because of them, and they made all of the discoveries that I didn't get to talk about and the discoveries I did, so that was really fun. This is my husband, Todd, who's supposed to be here today, but he has COVID. It's because he doesn't believe in microbiology. He has COVID, so he is at home. I have tried to tell him, and he's been my partner in all of this. And then I'll finish by showing a picture that Mike already showed you. So of course, the real reason I'm standing here is because I had the most fabulous postdoc mentor who taught me to be a scientist, who taught me to be a geneticist. And then in one of the greatest gifts that a mentor can give and that he taught me, Mike gave me my project to take to start my lab. And I have never worked on anything since then. And that is the highest form of intellectual fatherhood or whatever. And so I want to thank Mike and Jen, and I want to thank you and the Gairdner one more time. It's a real delight to get to give this talk.
Just absolutely fantastic, and I think, Peter, to your concern, it looks like this field has legs. So it's my pleasure now to introduce Dr. Abiola Olatan, who is a 2023 Gairdner Early Career Investigator, and he's going to present on how Clostridia difficile hijacks heme to mediate metronidazole resistance. Abiola Olatan is an assistant professor in the Department of Biology here in Canada at the University of Waterloo. He received his PhD from Marseille University in France, where he studied the molecular mechanisms underlying resistance to last-line antibiotics. And he pursued previously postdoctoral research in host pathogen interactions, specifically investigating the organismal processes of bacterial infection in C. elegans at both Duke University and Oregon Health and Science University with Professor Alejandro Abele. Afterwards, he transitioned to study the molecular mechanisms of heme-dependent metronidazole resistance in C. difficile and his global epidemiology at Texas A&M University with Professor Julian Hurdle. His research group is focused on understanding the molecular mechanisms of stress defense responses and antimicrobial resistance in C. difficile. Please join me in welcoming him to the stage.
Okay. Can you hear me? Okay. So thank you so much for that wonderful introduction. I'm very happy and honored to be here this morning, and I want to thank Gairdner for selecting me as one of the early career investigators. So this morning, I'm going to be sharing with you on some of my recent works, which I have titled, so how Clostridioides difficile hijacks him to mediate metronidazole resistance. And I would like to start with this story that we've all been hearing about quorum sensing. So with quorum sensing, the field of quorum sensing has indeed illuminated and shown us the extent of chemical signaling in bacterial communication and how it affects bacterial infection. But at the same time, we've also been able to also see that chemical signaling is also very much important in bacteria to host communication, which might also involve infection. And that has led, that's part of like what my research is also focusing about, not really on quorum sensing, but the interaction that exists between bacteria and hosts with respect to chemical communication. So in C. difficile, it was recently reported that C. difficile infection led to accumulation of heme in the gastrointestinal tract of the host, and heme is one of the products produced by the host. And at the same time, it was also shown that not only that heme is produced during infection by C. difficile, but it is also one of the virulence factors in C. difficile, because C. difficile mutants that is unable to cope with heme, or that is unable to detoxify heme, they lost ability to become virulent. And for my research, apart from talking about virulence factor, we have also discovered one of the importance of heme, which involves antibiotic resistance. With antibiotic resistance, we all know that it is one of the problems that we face in the 21st century. And in fact, according to a recent paper published in The Lancet last year, it reported that in 2019 alone, about 3,500 people died of infection related to antimicrobial resistance. And this is actually, we can say it's more or less equal to about 8 Boeing 787 crashing each day. So it is not actually a silent pandemic. It is indeed a major pandemic. And in fact, the paper also went further to say that in the year 2050, about 10 million people will die each year due to infection related to antimicrobial resistance, with a huge economic loss. So with regards to C. difficile, antibiotics that are available for the treatment of C. difficile, they are very few. In fact, we currently have 3: metronidazole, vancomycin, and fidaxomicin. Metronidazole has been one of the antibiotics that has been used for several years for the treatment of C. difficile infection, and it is very cheap. In fact, $21 compared to $1,400 for vancomycin. And over the years, it has been observed that metronidazole efficacy is not efficacious in patients. In fact, there have been high treatment failure associated with metronidazole therapy in patients over the years, as we can see here. However, the mechanism or the reason why metronidazole is failing is not known, and that has made metronidazole to no longer be recommended as part of the first-line treatment option for C. difficile again in several countries, including US and Canada. So one of the focus of my research is to be able to understand why metronidazole is failing in patients. And for this, we made a very interesting discovery in my research group during my postdoc, in which we were able to identify that heme not only mediates metronidazole resistance in C. difficile, but it is also very much important for the detection of metronidazole resistance in C. difficile. Here, in the absence of heme, we could see that in red, all the C. difficile that we tested, they display very low MIC to metronidazole. and which means they are sensitive. However, in the presence of heme, when we add heme to the medium, we could see that some subgroup of C. difficile actually displayed, or they have elevated MIC to metronidazole, meaning that only in the presence of heme, that is when they show resistance to metronidazole. And another interesting thing here is that about 41% of C. difficile that we tested actually display heme-dependent metronidazole resistance. And that means without the presence of heme, we would have characterized all the isolates as sensitive to metronidazole. Another interesting observation was that in C. difficile, we have epidemic, hypervirulent C. difficile, and historic one that are not known to really cause major infection. So in this result, we are able to see that most of the epidemic ribotypes actually display high level of metronidazole resistance as compared to the non-epidemic, non-virulent C. difficile that are mostly sensitive to metronidazole. So we also wanted to know one of the clinical implications of this observation. So we performed retrospective studies involving C. difficile patients that failed metronidazole therapy and those that were successfully treated with metronidazole. And what we found was that in this, among those patients that actually failed metronidazole therapy, they are highly likely to harbor C. difficile that are resistant to metronidazole or that showed elevated MIC to metronidazole, and these are all heme-dependent. So meaning that there is a correlation between metronidazole increased MIC and treatment failure in patients. And in fact, this is one of the first study. Oops, it's not working again. So in fact, this is one of the first studies to be able to link metronidazole resistance to treatment failure in patients. So we wanted to understand what is the molecular mechanism behind this heme-dependent metronidazole resistance. And for that, we created transcriptome mutagenesis library. And from the screening of that library, we were able to identify a mutant that was now sensitive to metronidazole. And that mutant, the gene was identified as 5-nitroimidazole reductase, which we call NIMB. And it is also very important to indicate here that the sequence of NIMB between sensitive and resistant strains, they are both identical. There was no difference. So we wanted to understand what is the reason why NIMB is actually causing resistance if sensitive and resistant all harbor the same sequence. So what we did was to perform genome-wide association studies that involve analysis of both a collection of resistant and sensitive C. difficile. And from that, we were able to actually identify that NIMB, metronidazole-resistant C. difficile, they've actually developed a unique mutation in the promoter of NIMB. And that actually activated the NIMB gene, which now led to metronidazole resistance in those subgroup of resistant. And that mutation was absent in all the sensitive strains that we've looked at. So I've been talking about NIMB and heme. So what are the relationship between these two? So finally, we were able to identify that NIMB is indeed a heme-binding protein with this characteristic peak that it showed, which showed that it can bind with heme. And also, we're able to identify the heme-binding residue in NIMB, which is at histidine 55. And when we mutated that histidine 55, we showed that NIMB lost ability to bind with heme, or it displayed a heme-binding activity. And also to see that the relevance of this heme-binding ability of NIMB, we express both the wild-type NIMB and the mutant NIMB in sensitive C. difficile background strain. And what we saw was that the wild-type NIMB that was able to bind with NIMB, so that was able to bind with heme, will actually confer high-level resistance to metronidazole in the sensitive wild-type. However, the mutant that has lost ability to bind with heme, did not or lost ability to confer resistant to metronidazole in the presence of heme. So this shows that the ability of NIMB to actually bind with heme is very important for NIMB to be able to confer resistance to metronidazole in C. difficile. And going forward in the lab, we are very much interested in being able to reverse metronidazole resistance by identifying drug molecules or adjuvants that are able to prevent this heme to NIMB to heme interaction. And if we are able to prevent this, then we'll be able to reverse metronidazole resistance in C. difficile. And not only in C. difficile, but in other bacterial pathogens, because NIMB is present in different anaerobic bacteria. And this will be very important because, going back to my story, that metronidazole is very cheap, even though it is no longer recommended again, but in several other countries, it is still the preferred drug, and it is also used in other anaerobic bacterial pathogens. So the ability to be able to reverse this metronidazole resistance will be very important in controlling anaerobic bacterial infection. And lastly, I would like to thank my mentor, Professor Julian Hodu, and several other collaborators that have contributed to the success of that study. And at the same time, I would also like to thank my colleagues that have been very much helpful during the course of my research. And lastly, the funding agency that funded the research. Thank you.
Thank you. It's my great pleasure to introduce our second early career investigator, Dr. Elena Guzman. Dr. Guzman received her undergraduate in human biology and zoology at the University of Toronto. She also did her master's here at the University of Toronto in the Department of Laboratory Medicine and Pathobiology, doing her research in the Sick Kids Research Institute. And then she progressed to the Donnelly Center under Charlie Boone and Brenda Anders, where she did her PhD studying multiple genetic determinants and how they combine to affect fitness. She has conducted pioneering studies on mapping complex genetic interactions in yeast and showed that their key role in the genotype to phenotype relationship. She did a postdoctoral fellowship at McGill University under Professor Morag Park, and as a postdoc, she showed that mutations in multiple genes drive cancer. She is currently assistant professor in the Department of Biology at Concordia University, and she is a Canada Research Chair Tier 2 in synthetic and functional genomics. Please welcome Dr. Guzman to the panel.
Thank you very much for the opportunity to present here. This is so humbling, such an honor. So I will tell you today about complex genetic interaction networks in yeast and human health. Let's switch gears a little bit from what we heard today earlier and talk about precision medicine. So the Canadian Cancer Society defines precision medicine as a form of medicine that uses persons' genes and the genetic and molecular profile of cancer to prevent, diagnose, and treat disease. And the goal of precision medicine is to go beyond general treatment regimens for patients to a more patient-centered approach. And ultimately, the key is to minimize adverse events while improving the effect of therapies. And so we think of cancer as a genetic disease that results from the accumulation of mutations. And typically what it's thought is driven by single gene alterations, either gain of function of oncogenes or the inactivation of genes, which we refer to as tumor suppressors. What's being appreciated more and more is that large perturbations that encompass simultaneous deletion or amplification of multiple genes, they're also important cancer, and they're frequently observed. And so these chromosomal arm aberrations or alterations that span entire chromosomes are referred to either aneuploidies or large copy number variants. And this is going to be the focus of my work. So a seminal study, pan-cancer study, now a number of years ago, led by Matthew Meyerson's group from Dana-Farber, showed that there's preferential gain and loss of very specific chromosomal arm changes. So what's shown on this quadrant here, for example, is that these chromosomal arm level changes are often seen as deletions across cancers. And they're rarely seen as amplifications, whereas these chromosomal arm level changes are shown often are observed as gains, but rarely as losses. You don't see many in this quadrant, suggesting that these are not stochastic events and that cancers really prefer these alterations in this specific way. And actually, a recent study that was published in Nature by Taylor and Barakim Labs actually showed evidence for a positive selection that are related to tumor fitness of these specific cancer aneuploidies. And so in the lab, we're focusing on breast cancer. And breast cancer is a heterogeneous disease thought of and categorized into three main categories based on biomarker expression. And so there have been targeted therapies devised against breast cancers that present with estrogen-progesterone expression, estrogen-progesterone receptor expression or HER2 amplification. But triple-negative breast cancer that lacks these three biomarkers lacks targeted therapies and largely is treated with chemotherapies. Recently, there has been advance and some success with immune therapy and androgen receptor inhibitors, but that targets and is applicable for a subset of these patients. And so the question is, how do we target these cancers? What is apparent, though, is that triple-negative breast cancer, despite lacking common oncogenic drivers, except for p53, which is not targetable, presents with large chromosomal deletions. And one of those deletions is chromosome 4p, which is highly recurrent, that I will tell you next. And so what we decided to do is to ask this question of how do recurrent large copy number variants evolve in triple-negative breast cancer, and what are the selection pressures that maintain them? And so we used a number of approaches. Sorry, I'm just going to come back here. We used a number of approaches to answer this question, utilizing the Cancer Genome Atlas data that's publicly available, as well as our own primary tumor patient-derived xenograft models to do phylogenetic reconstruction using single-cell and whole genome sequencing, as well as genetic screens. And I will show you a subset of those sort of findings. So chromosome 4p loss is broad and encompasses a large fraction of the chromosome arm, depicted here in blue. So the chromosome here from telomere to centromere, patient samples are oriented vertically, and on the x-axis are the coordinates. And as you can appreciate, patients present with an alteration and a deletion that spans a large fraction of the arm. And this chromosome arm level is functionally significant. So this, for example, shows that patients who have this chromosomal deletion tend to do worse. So they have a poor outcome and they have worse survival. We worked then with Peter Van Loo's group, who used to be at the Crick Institute and is now at MD Anderson, and has been leading the PCOG initiative, the pan-cancer initiative of whole genomes. And we worked with his group to really analyze the evolutionary history of triple-negative breast cancers using our own patient cohort. And what we've observed is that chromosome 4p loss is an early event in tumor progression, and it's marked here by this black arrow, and it's often observed early in tumor evolution. And so this is a leak plot. The alterations are ranked by how early they emerge in tumors. So chromosome 4p loss happens around the same evolutionary time as coding mutations in P53, which is the master regulator and a tumor suppressor, 17p, which carries P53, chromosome 5q loss, which carries RAD genes, which are important for DNA damage and repair, for example. And so through a number of analyses, and actually not shown here, but we've dated it back even to benign cancer that even seen before the malignant stage, chromosome 4p loss. So what we did is through a number of assays, and one of them is shown here, including a genome overexpression screen, we showed that chromosome 4p confers a proliferative advantage. So when we overexpress genes in the region, their overexpression inhibits cell growth. So it suppresses proliferation, which is shown here in blue. And one of the genes that is important for this effect is actually used to be an unknown gene, C4orf19, and it looks like we're on this path with other presenters to characterize genes and name them, and unfortunately this is not as fun as Q-tip. But C4orf19, it's called that because it's on chromosome 4, so C4, and orf19, it's orf number 19. And what we did is this orf, when overexpressed, suppressed proliferation of chromosome 4p. We mined bioGrid data, which is a curation of genetic interactions and protein interactions that's led by Mike Tyers, who is also an investigator here at U of T. It became apparent that it interacts with a very specific submodule of a larger stripe-back complex. And it involves, which has been known to affect cell growth and regulate cell death. And the submodule is PDC-D10, programmed cell death protein, and serine threonine kinases 24, 25, and 26. We then collaborated with Enkla Jingara's lab, and her lab is also here at Mount Sinai, and showed with CoA immunoprecipitation experiments and biotinylation proximity analysis assay that these, in fact, C4orf19 interacts very specifically with this submodule and no other component of the stripe-back complex. So it really looks like it's competing with striatins for binding to PDC-D10. It seems to carry its function at the plasma membrane, as it's apparent that its proximal its interactors are in the cell periphery, and the immunofluorescence analysis shows its localization to the cell membrane. And so what we think it's doing is that C4orf19 is binding to PDC-D10, which is then bringing GCK kinases to their substrates at the membrane. To regulate cell proliferation and regulate cell death. What also became apparent to us is that the suppression of proliferation was also context-specific. And in fact, if you look underneath here, this reflects the results of the genome-wide overexpression screen in also the cancer in a triple-negative breast cancer that carries chromosome 4p deletion, whereas the initial results I showed you were in a normal breast epithelial cell line. And so what it shows is that if a triple-negative breast cancer is evolved with to contain chromosome 4p deletion, they will be much more sensitive than normal epithelial cells to overexpression of genes with this region. This was not only specific to chromosome 4p, but as was also apparent for other regions, such as, for example, 13q, which is also deleted and commonly deleted in these cancers. So it likely presents a mechanism that selects for these chromosomal aberrations and logs them into this copy number state. The other finding that we saw is also that the PDC-D10 GCK3 module, the members of these module are along these chromosome arms that are lost and are lost early in tumor progression. So maintaining stoichiometry of this protein complex is another mechanism that's likely is important for the evolution of these tumors. And so with that, what we're doing now is that we are looking for targeted therapies. So how can we target chromosome 4p loss? What other combinations of genetic perturbations will selectively kill these cells? How genes, we're doing a combinatorial genetic interaction screens to see how do genes within chromosome 4p combine to affect cell growth and to affect fitness. And this is along the line of my PhD work in Charlie Boone and Brenda Andrews' groups, where we did systematic analysis of complex interactions in yeast. And I'm continuing also and have some yeast projects in the lab where we're looking at genetic network rewiring. And so how these complex genetic interactions are conserved from yeast to worm to human, and how they rewire, and how can we learn about the genotype to phenotype relationship that's so important for developing novel precision medicine therapeutic strategies. And with that, I would like to thank the members of my lab, my postdoctoral supervisor, Morag Park, and our collaborators who already mentioned, and my PhD supervisors, Charlie Boone and Brenda Andrews, who really ignited the curiosity and passion for studying these complex genetic interactions. Thank you very much.
Okay, our next speaker is Dr. Gilia Zada, and she is the winner of the Gairdner Momentum Award. She will present on integration of molecular information to advance patient care in real time. Dr. Zada is the Dan Family Chair and Professor of Neurosurgery at the University of Toronto, and this role makes her the first Canadian Chair of Neurosurgery. She is the head of the Department of Neurosurgery at the Toronto Western Hospital and medical director for the Krembil Brain Institute at the University of Health Network. Dr. Zara is senior scientist at Princess Margaret Cancer Center, where she runs a translational research program, and she holds the Wilkins Family Brain Tumor Research Chair. She has held several leadership roles and organized neuro-oncology and neurosurgery societies nationally and internationally. Please join me in welcoming Dr. Zara to the stage.
Thank you very much for the opportunity to be here, for the recognition. Congratulations to all the other award winners for the Gairdner this year. And I really want to recognize Dr. Janet Rossent for her vision for introducing the Momentum Award, which I'm the inaugural recipient with my co-recipient, Christopher Mushbosh. So it's really great to be here and to show you some of the work that we've been doing on brain tumors. I'm a neurosurgeon, and my clinical practice focuses mainly on neuro-oncology, skull-based tumors, and so my research parallels that. I have no conflicts to disclose, just to tell you that this year is 100 years of anniversary for neurosurgery at University of Toronto, and we will be celebrating that in May. If you look at the list of World Health Organization that lists the number of brain tumors that we can have, it's about 150. This is diagnosed by our neuropathology colleagues under a microscope based on histopathology. However, these are not all of the tumors that we deal with on a daily basis in our practice. These are the main tumors that I deal with in my day-to-day practice. Pituitary adenomas are benign tumors that stem from the pituitary gland that sits here. It's a kidney-shaped organ that controls all the hormones in our body. It's vital and essential for us to function. The tumor continues to grow. The optic nerves typically should be sitting here. The tumor compresses the optic nerves way up high, and this red is the tumor. Advances in technology, in particular visualization and endoscopy, allows me to avoid having to do an open operation and convert this to a minimally invasive endonasal through the nose. I can remove this tumor completely. The patient is cured, their vision is restored. Very rewarding for myself, but more importantly, obviously, for the patient. Another tumor that has benefited significantly from advances in technology is vestibular schwannomas. Tumors that stem from the nerve that go to our ear that allow us to hear result in loss of hearing. I think the pointer's not working. And in the past, we would have had to do this by surgery. However, we now have converted this to a procedure where there's focused radiation delivered to the tumor and spares the normal brain. The tumor rate control is about 90% and avoids operation in majority of these tumors and only restored to tumors that are large in size and volume. The tumors that are growing in size, or the ones that you could see in the middle panel, are the meningiomas, and I'll talk to you mostly today about them. And these are tumors that somehow always pose a challenge to us as surgeons because they wrap around neurovascular structures, and resecting them is challenging. The tumor that majority of you have likely heard are glioblastoma, where at the time of presentation, you could see on the MRI, there's the red line that delineates this tumor. We can tackle this tumor by surgery and remove it safely for the patient. The challenge is the green line here. Tumor at the time of presentation is invaded significantly beyond where we could see the mass. And so, of course, we can't resect that completely. And these invasive cells that you could see as the black dots on these post-mortem images is what causes the challenge of tumor recurrence, and the patient population don't do very well. So regardless of the variety of chemotherapeutic trials that we have for glioblastoma, you could see that the average survival is around 9 to 12 months. Overall survival, unfortunately, is less than 24 months, and the tumor always comes back. So we need to do better for this patient population. Meningiomas that I'll show you also, although majority of the time do well, have a subset that do extremely poorly. This is in years by comparison to the GBM. What are the treatment paradigms that we offer patients in neuro-oncology? I would say it's rather linear and simple. The patient presents with a symptom, weakness of an arm, difficulty with speech, seizure. An MRI is done. We then either choose to operate because of the appearance of the tumor being concerning, or if not, we undergo surveillance imaging until the point the imaging characteristics are concerning, and we go to surgical resection, optimizing maximal safe resection where we can. We wait for about a week to 10 days for pathology to come back. There are some molecular features that we integrate into our diagnosis. And then unfortunately, this is where we have a gap in our care. We go to a very standard chemotherapy and radiation, and often patient fails those, and we have to go on to better treatments for our patients with brain tumors. Histopathology has been the mainstay of diagnosis, and if you look at this section, to the untrained eye, it's very easy to see that this is likely dead, hypoxic, necrotic tissue. These are hyperproliferating cells creating what's very characteristic for a GBM, a pseudopalisading hyperproliferative area with endothelial piling. These structures are abnormal vasculature. This is a glioblastoma. So even us who don't do neuropathology can make this diagnosis. Other characteristics we look for, fried egg appearance. This is an oligodendroglioma. Of course, we've advanced, and our understanding of gliomas in particular rests more with classification based on molecular alterations to layer on top of histopathology. If you look at this section, to a neuropathologist, to myself, this looks like a normal brain. However, I draw your attention to the circles. A defining molecular alteration in gliomas is the mutation of IDH, which is an enzyme, and that mutation results in a better outcome for glioma patients, so they have a better prognosis. If you stain for IDH, I want you to take note that only a few cells here stain positive, telling us that this is not normal brain. This is a low-grade glioma that has an IDH mutation, and it's the start of a glioblastoma forming in this person. But note that also IDH is not positive in all of the cells. This becomes important in a second with what I want to show you. So we've moved on from using histopathology to characterize glioblastoma, gliomas, brain tumors in general. We've layered some of the molecular knowledge that we've gained over the last 2 decades into better classification of brain tumors. IDH mutation, as I showed you, being one of them, 1p19q being the other, and then the question is, what does this matter? It matters because if you have a low grade that is an IDH mutant, your outcome is very different from a glioblastoma that is IDH wild type. It bears on how we counsel the patient. It bears for me as a surgeon to decide how aggressively to pursue extent of resection, and ultimately, hopefully, it bears on treatments, which we have this year, in fact, seen that IDH inhibitors are effective for this population. Another evolution in our understanding is the seminal paper that was published in 2018 by the DKFZ group in Heidelberg, which showed that if they took approximately 3,000 brain tumors from 80 different subtypes and did methylation analysis on these tumors, the methylation signature is very distinct, and it's akin to a fingerprint where IDH mutant and IDH wild type separate out and all the other tumors. And of all tumors they analyzed, there was a 15% change or improvement in accuracy of diagnosis, arguing that you must layer on top of your traditional histopathology methylation to be able to give the patient a better diagnosis. What we did in our group, and I have to give credit to Ken Aldape, who was the neuropathologist at the time here, introducing methylation in parallel to the DKFZ group, and we looked at our results in our institution here at UHN, and you could see that the initial pathological diagnosis based on histopathology is on the left. The integrated diagnosis incorporating the methylation that I showed you is on the right, and the degree of crossover is significant. We did this in part because our funder, the Ontario Health, told us, well, what's your data to support improvement in outcome for the patient, so we would fund this. And so we looked at all of those that had a clinically challenging case where, for example, 2 or more pathologists couldn't reach consensus. The diagnosis given by the pathologist did not match what we thought it was clinically, in particular what we see in the operating room. Molecular features could not be ascertained using molecular testing. And of all those cases, you could see that 84% had a meaningful change in their diagnosis. 24 had a change in diagnostic entity, so where they were called a glioblastoma, they in fact were brain metastases. Significant difference for that person as to what you would do with them. 31% had a resolution of their differential diagnosis where 2 or more pathologists couldn't reach consensus, and 27% had a change in grade. So where we would have previously said it's a grade 2, observation would be the next step. Grade 3 or 4 means patient needs radiation or chemotherapy. The next question is, what does this mean for the patient? And of all of our population that we reviewed in that 5-year period, 15% had a change in clinical management, a significant number for what we do for our patients day to day. We now use this on a weekly basis in our tumor board. The next area is this young man comes to the emergency department with seizure, focal seizure of their hand, and MRI is taken, and you could see even to the untrained eye, this is the abnormality. This is a profile view of the person, the brain folds. This abnormality sits right up against the structure that's called the motor cortex that control gross movements of our arm and leg. The supplementary motor area is in the front of it, and it's actually the command center for me to convert crude movement to sophisticated movement, as sophisticated as playing a piano, as sophisticated as me speaking to you today. So if I were to take this out, the person is at risk of having some issues with their supplementary motor cortex. And when I go into the operating room, though this is very obvious to you here, it's not very obvious in the operating room. So you don't want me to explore to find this tumor. The next image is a live image of a brain. So if you want to look away, if it bothers you, please do so. Because when I open the brain, this is the correlating image, and it all looks normal. These are the normal folds of the brain. These are the arteries. These are the veins. We have end vessels in our brain called perforating and microvasculature. It's amazing. It looks beautiful, but I can't tell where that abnormality is. So I need adjuncts. But I also need to use my molecular knowledge to help this individual in this treatment paradigm make a decision for that person better. What can we do? We know that tumor cells shed small pieces of circulating tumor DNA into the bloodstream. And we can pull down those circulating tumor DNAs and capture the genomic information on them, whether it's mutation or methylation. Mutation is challenging. If you recall, I showed you that the IDH mutation is only present on a few cells of a brain section. So you can imagine that if the tumor cells are shed, they're not going to all have that IDH mutation, and therefore my detection rate is going to be low, and the value of that is going to be limited. However, methylation, which I showed you is now used as a classification, is conserved on all pieces of DNA. And this is where the beauty of working in a place like this at Princess Margaret comes to play, because I work with Daniel de Carvalho, who published in Nature in 2018, showing for all other cancer types that he can use circulating tumor DNA, their methylation signature, to discriminate between a range of systemic cancers. Leukemia, of course, is a liquid cancer. It's 100% accurate. Breast cancer, renal cancer, lung, pancreatic, kidney, all with some degree of accuracy, you can discriminate between them. So we explored this in the brain, and you could see that with a very high accuracy, you can discriminate gliomas from all other. We didn't anticipate this because there is this thing called the blood-brain barrier, which avoids material going to our brain and vice versa. We felt maybe, in fact, that's not going to give us the ability to detect these circulating tumor DNAs, but it does, and it allows us to discriminate it against other cancers with relatively high accuracy. The IDH mutation status can be also discriminated, where I mentioned to you IDH mutation for the person that came to emergency that I showed you the MRI makes a big difference, because if their outcome is good, the progression of the tumor is low, I will back off on doing an aggressive operation on that person. Versus if I knew that it's an IDH wild type, likely becoming a GBM, I would operate on that person sooner. The next is, well, maybe in grade 4, those endothelial cells that I showed you that are piled up are disrupted, the vessel is disrupted, DNA is shed better into the bloodstream. You could detect grade 4 better than the other grades, but in fact, you could see grade 2 and 3 compared to grade 4, equal accuracy. So I think we have a test that can make this diagnosis. The next question is the signal we're picking up on the circulating tumor DNA coming from the tumor cells, the tumor microenvironment, or other factors? Majority of our patients are on dexamethasone. Are we altering the signature of the white blood cells that we're detecting? We layered the methylation of the blood onto what you could see the methylation signature of a number of cell lines. We then layered that onto what we have in the plasma, and you could see, in fact, that with very high accuracy, it discriminates it for a glioma-specific methylation compared to other cell lines. And so we have a test that with accuracy can allow us to layer on top of imaging, not by itself alone, and improve the accuracy with which we make a diagnosis and then subsequently a decision for the patient. If you look on the left, this tumor, this and this, to your eye and my eye also, look very similar. I can't tell if this is a meningioma or a hemangioparasitoma. Totally two different tumors, very different characteristic, very different surgeries that we would plan. In the case of a hemangioparasitoma, you could tell that the blood test very accurately, almost 100%, with no spill into the other columns, can tell me that that's a hemangioparasitoma. The next one, meningioma, looks pretty identical to this one, to the trained and the untrained eye. This one is a meningioma, a little bit of spill into the others by the plasma test, but really with very high accuracy, allows me to plan for surgery better, and so on and so forth. So similarly, you could see as we get into the more malignant gliomas, the accuracy decreases, but we have imaging to layer that on. So this was published in Nature Medicine. I credit all the authors. These studies take multiple collaborators, multiple individuals to make them happen, and really its credit goes to all the individuals who did the work. Coming back to this patient, I would then submit to you that if I had this image, the young 29-year-old in the emergency department, high-risk area of surgery, if I do a liquid biopsy that tells me it's an IDH mutant tumor, I would be conservative in wanting to pursue an extent of resection that's aggressive, and we would monitor that person. So hopefully I've convinced you that with some layering of our molecular knowledge, we've added and made more sophisticated this treatment paradigm. Next are meningiomas. These are tumors that I could potentially have in my head, and it just never grows. A lot of us on postmortem have these small growths. They come from the covering of the brain. In fact, they're the majority of our brain tumors, not glioblastoma, though the field focuses on improving the outcome for glioblastoma because they do so poorly. Meningiomas are the most common tumor, and the incidence increases once we hit around 55 compared to the other brain tumors. Histologically, they're graded into 1, 2, 3. Majority are grade 1, and majority are benign. Majority can be resected by surgery, and the patient is cured of their condition. However, grade 2 and grade 3, unfortunately, cannot be cured by surgery, and postoperatively, we're very limited in what we can offer the patients, and this is why we did the studies. If a patient presents with an incidental, what looks like a small meningioma, these are not the same two patients, it's hypothetical. It's difficult to say at the time you're diagnosed and you come to see myself or someone that this tumor isn't going to become this size versus this tumor stay the same for years. There's no way of predicting it other than to keep repeating the MRI. There have been attempts to try and better understand the molecular biology of meningiomas, and about 2 decades ago, there were 2 large-scale studies that looked at the mutational profile in hopes that, similar to IDH mutation for meningiomas, we can find a defining mutation. But you could see that, unfortunately, in grade 2s and grade 3s, other than NF2, there are no recurring mutations. The mutations in grade 1 have been targeted in clinical trials, but these are very slow-growing tumors, so the results are not out. There is an increase in copy number alteration due to the disruption, as our previous speaker spoke, of copy number alteration with increasing aggressiveness of the tumor and a slight increase in TERT promoter mutation. However, I want to draw your attention to something. Grade 1 meningiomas in these studies were 500 or 600. That's the total number we get per institution, roughly, per year. Grade 2, however, is 270 less. Per institution, we would not get 270 per year. Grade 3 are only 15, so significantly underrepresented. What do we do when cancer is rare and we have underrepresented samples in a single institution? In my opinion, you need to bring many institutions together, which isn't easy. So creating an international consortium of meningioma is something we set out to do in 2015. 2016, we launched it. We published books, journal manuscripts, articles together so that we increase awareness and the need to focus on research and, more importantly, share data. So it made possible for us to then create cohorts of tumors that are represented by grade across various institutions so that we could do a more accurate study. If you look at methylation, which I mentioned to you, of all meningiomas, you could see that they divide into 2 groups, a test and a validation, a Princess Margaret and an MD Anderson cohort. There's a favorable outcome group and an unfavorable, regardless of the WHO grade, whether it's grade 1, 2, or 3, regardless of the age, regardless of the location, extent of resection, or any other factor. By methylation alone, they divide into favorable and unfavorable. We published this, and at the same time, the DKFZ group, who I showed you the methylation data from, took all their meningiomas and looked at their methylation, and they divided it into 6 groups. But I would argue the favorable is the first 3, the unfavorable, the last, and the intermediate. They layered some of the mutational changes that they saw. But regardless, these 2 studies corroborated independently, telling us that there's something in methylation in meningiomas that tell us the outcome of the patient, independent of any other clinical parameters that we use. So we then went to the process of trying to create an individualized nomogram or a calculator for a person. So for me, what is the risk of a 5-year recurrence of my meningioma? Again, going back to this, is mine going to look like this or going to stay stable? In order to do that, we took a cohort of meningiomas from 2 institutions, Heidelberg and here, Princess Margaret. We analyzed them with methylation. There are 10,000 probes on that methylation array that distinguish the favorable from the unfavorable. We used that 10,000 probes to create a pipeline using computational models to be able to then distinguish one meningioma from the other based on the presence of that 10,000 methylated probes. And then we repeated this in multiple cohorts. For the purpose of the publication, we repeated an additional 2 cohorts. And we layered clinical factors that we use currently, the grade, the extent of resection, age of the patient, gender, et cetera. And we came up with a modeling that can then supersede all of our other clinical criteria. In all WHO grade 1s, so these are tumors that we know historically are very good behaving tumors, but in our practice, we have meningiomas that are grade 1. I completely resect them, and the patient has a very fast recurrence. When you analyze it by the methylation pipeline that I showed you, yes, majority of them do well, but in fact, there is a subset that behave aggressively. And this is where the methylation pulls out that subset, and this is the population we need to study closer, and more importantly, offer them something other than surveillance imaging. Grade 2 meningiomas are very challenging and controversial. Some believe you have to radiate them right after resection, even if you had a complete resection, to avoid recurrence. Some say to continue surveillance monitoring. When you apply the predictor, you could see that there's a subset of grade 2s that do very well. So we would argue, and currently in practice in our tumor board weekly, we would then defer radiation for this population because they are going to do well. There's a subset that are intermediate, but there is a subset of grade 2 meningiomas that do extremely poorly, and so we need to focus on this group. Grade 3 meningiomas are considered to be anaplastic, aggressive. The duration of their longevity from the time of resection is typically 2 years. And all the time, patients receive radiation after resection. However, they may not have a benefit from that radiation. But more importantly, in all of our practices as neurosurgeons, we have grade 3 meningiomas that don't have a recurrence. And so this methylation can pull out this subset. And I would argue that this group actually doesn't need radiation. They could continue monitoring. It's this subset we need to look at. So how does the calculator work? This is the first calculator that we've created for any brain tumor type. It has 100 points weighed equally for the methylome. The grade of the tumor, the histopathology, is weighed at approximately 50%. Extent of resection is something we use as surgeons. We always overestimate our extent of resection, but it's calculated on imaging. And the score that's given to us gives us a 5-year predictive score, or a predictive score for a 5-year recurrence. So this patient would have a 0.4 score, a 60% chance of recurrence in 5 years. So I would say intermediate. These are some examples. This is a meningioma. It's a convexity-based. It was resected elsewhere. Good resection, but you can see that there's residual. The grade of the tumor is 4. The Simpson grade is, the grade of tumor is 2. Simpson resection is 4. The methylome score, based on analysis of the methylation and the pipeline that we created, is approximately 0.09. The inverse of this is the likelihood of recurrence, and you could see in 33 months this patient had a recurrence. By comparison, similar type tumor, slightly different location, similar type resection, the grade of the tumor histopathology is 2, extent of resection by the surgeon is 4. The methylome score, however, you could see is 0.77. The inverse of this is the likelihood of recurrence, which is much lower than the last case. And you could see 96 months later, this person has no recurrence. We now have a wealth of cases such as this that we've managed and analyzed by methylation scoring to know that, in fact, the pipeline is accurate, and we apply it to daily practice. I was partnered with Dr. Belazan, one of the other graduating, Gairdner Awards. You've graduated some time ago. And the question really is, from a discovery, when I tell you with great enthusiasm, show you the data that's compelling, how long does it take to adopt that into clinical practice? And in my naive mind, I thought I'll show this a few times to my radiation oncology colleagues, and away we go, we use it. It took about 2 years to convince them, for good reason, for patient safety and the quality we need to ensure, that they now adopt this, and on a weekly basis, they actually ask of us for the methylation score. Some of the team members in my lab who do this are here, and so we give them the score and we make a decision for the patient. Absolutely rewarding cycle of translational research. So the next question is, we're at the peak of the wave of genomic era, in my opinion, in cancer, but I could be wrong. However, we have a wealth of data on genomic analysis of tumors. If you take a cohort of meningiomas, 120, that have equal representation of 1, 2, and 3 by grade, and analyze them by different genomic platforms, the question is, do they all fall into the same membership based on the platform you analyze? Because which one do we apply in clinical practice? And the answer is actually no. When you take 120 tumors and analyze them by RNA expression, you could see 6 distinct clusters and 6 distinct outcomes. So that's great, because I've just shown you information that based on RNA expression, this tumor may have differences in outcome. If I take that very same number of tumors and analyze them by methylation, they also divide into 6 subtypes. However, the methylation outcomes are slightly different from RNA. And then if you look at copy number on mutational data, you could see that there also are 6 different groups, 6 different outcomes, but these memberships are not the same. You could see the degree of crossover. So if you take membership in methylation in group 6 It is not the same as membership in group 6 of RNA, and definitely not in copy number. So me as a clinician says, well, I don't know, which one am I supposed to use? Which one is more accurate for the patient? And we believe neither of them. You have to integrate them until you come up with stable clusters and groups. And so we took an approach of cluster analysis where we layered the various platforms of genomic data we have on the same cohort of patients, and you repeat this until you get stable membership. And you repeat it enough with different cohorts, validation cohorts, independent cohorts, so that there are 4 independent groups. And you could see that each of them are defined by whether it's methylation RNA copy number to a different extent, some more based on methylation than the other platform, but it's the same stable 4 groups. It's independent of the histopathological grade, it's independent of location, it's independent of the extent of resection by myself as a surgeon, and other factors. So the next question is, does it mean anything? And yes, because they have 4 distinct outcomes. MG1, meningioma group 1, has a better outcome, meningioma 4 has the worst outcome, and the 2 in between. The COCA, or the cluster of cluster, the molecular group analysis performs better. It's almost closer to a 1 than WHO grade, and better than that methylation pipeline that I showed you. So then the next question, obviously, is what is the biological meaning of these 4 groups? Are there pathways that are specific to one versus the other? And if you look at the RNA expression of the 4 groups, MG1 through 4, you could see that MG2 is a unique group. NF2 is not actually altered. I showed you earlier that on mutation, all of the meningiomas have NF2 mutation, majority do. This group doesn't, so they're unique. MG1 is immunogenic. All of the immune pathways are upregulated. MG4 are hyperproliferative, cell cycle proliferation, et cetera, pathways are upregulated. MG3 are hypermetabolic angiogenic. Those pathways are upregulated. So it tells us that, in fact, maybe clinical trials fail because we enroll all meningiomas equally and treat them the same, but give them one drug and say, oh, it failed. So we need to then design clinical trials that for immunogenic, potentially we use immunotherapy. For metabolic angiogenic, we use anti-angiogenesis, cell cycle inhibitor for MG4, et cetera. When we do pathway analysis, we can also identify drugs that are in existence that could potentially target these pathways. And I want to just focus on one. We've identified a number, but MG4 responds to vorinostat. And so we took cell lines from each of these 4 different tumor types, MG1 through 4, grew them in a dish, and looked at the characteristics of them. And I'll show you that in a second. The next big question, of course, is it's great to do all these platform analysis, but what happens to a day-to-day laboratory in a hospital that relies on immunohistochemistry? And the first step to make this accessible so that the 4 groups can be identified by an antibody that can then be stained in a histopathological lab is we do proteomic analysis. Of course, the proteomic analysis gives us a wealth of other data, but for the purpose of being able to convert it into immunohistochemistry, you could see that the cohort analyzed by proteomics divides into 4 groups, the same 4 groups that I showed you. And for each group, there is a distinct marker that we could stain by immunohistochemistry, where with high accuracy, it correlates to that group. This doesn't mean I can now use S100 to detect MG1. That's a second phase that follows from this study, where blinded pathologists review all the S100, and we in turn do the platform analysis. I don't think Aaron Schimmer is here to give me money for that, but I'll ask him. It never hurts. So the cell lines, MG1, 3, and 4, MG2 that are the NF2 intact do not grow in a dish nor in an animal, obviously. But they grew in a dish, and we treated them with vorinostat and 5-azacitidine to see whether there's a differential in the response of these lines. As I showed you, there are different natures to these tumors. And you could see that MG4 responds best compared to the other 2. 5-azacitidine doesn't impact any of the lines. Similarly, in mice, the tumor grows smaller, and then the survival is longer when treated with vorinostat only on MG4 lines and not others. So this was published in Nature in 2021. Again, very large study, multiple authors, many in my lab, many collaborators across the world, in fact, where we reached out to be able to address the questions the reviewers had. And in summary, I hopefully have shown you that we can move away from histopathology, not to abandon it, but to layer it and use molecular information to divide meningiomas into 4 groups. This bears on how we design clinical trials moving forward to show efficacious therapeutics for the population that we need. They have somewhat propensities of different location in the brain, very distinct outcomes, very distinct methylation patterns, copy number alterations. We've identified immunohistochemical stains. And at a single cell level, data that I haven't shown you for the interest of time, we could see at a single cell level the degree of variability in the 4 groups from each other, but really relatively adherent to what we see biologically within each group. So again, I really want to thank you for the opportunity. This is a tremendous honor and recognition for what I do day to day. All of the granting agencies that support all of the work that I've shown you. But more importantly, I really stand here for the team that does the work and represents us as a group, my lab, all of the members that are current, past members. I have to work on capturing all the names, and all of the collaborators that we have here and outside internationally. Thank you very much for your attention, and there's no questions, I think, at this point. Thank you.
Okay, we're going to take a short break now. Help yourself to coffee outside the auditorium. Washrooms are just located down the hallway, and please exit to your right.
We're going to move on. We have a busy day ahead. Our laureates have a busy day ahead, and I would like to get them through this session, and I'd like you all to hear the great talks to come. So please hurry up and take your seats. And I'll sit down now. You can come back.
Okay, while everyone's seated, I'd like to introduce our next speaker. It is Dr. Christopher Musquash. He is our 2nd winner of the Canada Gairdner Momentum Award, and he will be presenting on community-led Indigenous mental health and addiction research. Dr. Musquash is an Anishinaabe and a professor in the Department of Psychology at Lakehead University and the Northern Ontario School of Medicine University. He is a clinical psychologist. He is chief scientist at Thunder Bay Regional Health Research Institute. He is the director of the Centre for Rural and Northern Health Research at Lakehead University. He is a Canadian Research Chair in Indigenous Mental Health and Addiction, and has expertise in rural and northern clinical practice and the development of culturally appropriate interventions for mental health and addiction difficulties in First Nations children, adolescents, and adults. He is a researcher, a clinician, and a First Nations scholar. Please join me in welcoming Dr. Musquash to the stage.
Thank you very much. Always nice to have the applause out of the way at the beginning. That way, that part's done. Tremendous honor to be here. Thank you so much. Thank you to the Gairdner Foundation, Dr. Rassant, for your vision in identifying and honoring mid-career researchers. It's a tremendous honor to even be considered for such an award. And after hearing the research that we've heard so far today, I hope that everyone here is really feeling as inspired as I am. And now for something completely different. So again, thank you so much to everybody, and I very, very much appreciate the opportunity to be here. So this is me in a sport jacket. I have a number of different jobs, and the way that I think about things is that I kind of have the same job. So even though I'm a clinical psychologist, I see patients a day and a half a week, and I have a research program, and I'm a VP research and chief scientist, and I have a research center. Really, my goal is to try and improve systems and services for Indigenous people, but also people living in rural and northern regions of the country. And so that includes a number of different things. It might include research, it might include clinical provision, it might include policy development, policy recommendation, and so on. So I really had a great privilege of being able to do a lot of things that overlap with each other. So even though it sounds maybe perhaps in title a little bit disparate, I see it all as the same thing. And really, you know, I think the one thing that where all of the Gairdner Laureates start is with some type of story, some type of story that brings us to where it is we are now. And so I'll tell you a little bit about my story. This is where I grew up. This is a town called Sulacud. It's about 4.5 hours northwest of Thunder Bay in northwest Ontario. For those unfamiliar with that part of the province, it's just north of Barrie, by quite a ways. And so it's an interesting place to grow up because it is really quite isolated. It's about 45 minutes, 50 minutes north of the Trans-Canada Highway. But it does serve as a hub for First Nations communities, remote communities, flying communities, all in northwest Ontario. And so it was from here where I really noticed a lot of difficulties in community with respect to mental health and addiction, both within community. My own First Nation, Pagwashing First Nation, which is Pays Plat, which is on the North Shore Lake Superior, a little ways away from here, near Thunder Bay. And also where I learned aspects of my culture that I was perhaps unaware of, that other people didn't have those experiences. And so I'll tell you a little bit about it. I was a bit of an anxious child, physiologically anxious. And for those of you who are, you know, who work in those fields, you'll understand that mental health difficulties, you know, can sometimes have different sort of symptom presentations. Sometimes really quite cognitive, sometimes really quite physiological. Most often a combination of both. And so I came from a long line of warriors and other things like that. And so when I was a little kid, you know, you sort of had these experiences of being plagued by stomach aches and nervousness and so on, okay? So the question I always get asked then is, well, how did I become a psychologist? And there's a lot of ways to answer that question. The first way to answer it is, well, I did a PhD in clinical psychology, and then I got licensed, and that's how I became a psychologist. But I mean, that's not a very satisfying story. People generally want like a much longer story. And the more and more I think about it, the more and more I understand. The reason why I'm a clinical psychologist is because when I was about 6 or 7 years old, my dad, he sent me off kind of in the bush by myself. And so there was a place where we used to hunt. And there's a spot where the trail breaks off into two paths. And so one day we're going here, and I asked him, you know, where does that one go? And he said, well, go that way. And I said, well, by myself? Yeah. He said, go that way by yourself. And off I went, and I was terrified. So if you've ever been in the bush when you're terrified, you know, you feel your head sort of pounding, and you're sweating, and you can hear your breathing, and there's like a dinosaur crashes through the bush, but it's a squirrel. Like you've had that experience, right, where everything's just heightened and terrifying. And I get to the end of where the trails meet up again, and of course I see my dad's orange vest, and it's way down the path. He didn't wait for me. I caught up, and I was probably crying, and I was probably pretty upset about that. And I probably got home and had to explain to my mom what had happened. And the next time we went hunting, we got to that same spot, and I thought he's going to do this again, and he did. And the next time we went, I thought he's going to do it again, and he did. But by the 4th or 5th time we got to that spot, and I said, I'm going to go this way. And I went that way on my own, and I wasn't anxious about that anymore. And I think that's why I'm a psychologist. And the reason is because many, many years later, when I learned about clinical psychology and treatments for all manner of disorders, the thing that I found out was that exposure is like the evidence-based treatment is at least a part of the evidence-based treatment for almost everything. So you have to go through the experience. You have to develop, and a number of things have to happen. One, your physiological response to repeated experiences starts to decrease such that you're able to cope with it better. But you also learn something else in the process, and that is self-efficacy, and that is your ability to believe that you're able to make the change that you need to make. And those are very, very important parts of any type of behavior change, but in particular coping with any type of difficulty. And so here I was, this young person, you know, growing up in a town where I didn't know anyone who went to university. I didn't know about any of the jobs that I hold now. In fact, I often say that I accidentally became all these things I never knew existed. And I think that's the reason, because throughout our career pathways, what you begin to see is that there are these spots where you have an opportunity, but you don't really know if you have the capacity to be able to do it. And if you've only ever had experiences where you've not taken the opportunity or been given the opportunity, been encouraged to try that thing and then succeed or fail and cope with that outcome, then the likelihood that you're going to go try novel things starts to decrease. And so I think this is kind of one of the places where this research story begins to start. So, I'm 44. You can think about that as being, somewhere in Sioux Lookout in 1985, somewhere like that. The other thing is I'm really interested in relationships. I was fascinated by the talks today because really everyone talked about relationships between, bacteria, relationships between different markers of different cancers and so on and so forth. And I'm really quite interested in relationships between people, but also relationships between people and the environment, really interested in relationships between constructs and variables. And one of the things that I'm really interested in, again, sort of coming from a First Nation background, is this idea of intergenerational trauma and the relationship between intergenerational trauma and the broader colonial imposition, which, there's a number of policies and things that have occurred, and you can begin to think about what those might be and how those contribute. And so thinking about those relationships, you begin to kind of try to figure out how it is that things work. And for me, I'm also really interested in specificity. So if I say that, for example, residential school has led to intergenerational trauma, I can say yes. I mean, that's true. And the very good thing about that is that more and more people are becoming aware of those things. So it's something that people are more aware of now than I think ever before. In fact, we heard an acknowledgement here today in terms of the traditional territory of the people who were here before the City of Toronto was here. And that's an important thing to kind of recognize, but the challenge is that I'm also a scientist on the other side and a clinician, and my ability to intervene then is kind of limited by the specificity of the model. And so the last residential school closed in 1996. So if you're looking at an independent variable, a dependent variable, or predictor and outcome, now you don't have a predictor, but the outcome still exists. And so that means there's a level of specificity that you've not delineated in your model, which makes it difficult for people like me who are working day to day, trying to help support people who have this syndrome, right? The other part of the difficulty is that the syndrome is a little bit non-specific. And so if you say intergenerational trauma, what is that, right? It's kind of a broad syndrome that can include a lot of different things. It's sort of like asking, what's a city? Well, I'm from Thunder Bay. Is Thunder Bay a city to you? Probably not. There's probably neighborhoods the size of Thunder Bay, right? What defines what's a city? Is it purely population? Is it whether or not it services other areas? Is it whether or not there's an IKEA? Like we don't really know if what the definition of that is. I mean, there's fields that look at these things, but again, the question of specificity is an important one. And so I'm very think a lot about what it is that can be the underlying mechanisms of action when it comes to things like what we're seeing going on in our communities with mental health and addiction difficulties. Now, it turns out that there's a number of ways to think about it. And there's a number of fields of study that also work hard to kind of delineate and clarify these things. And one of the areas that I think is really an interesting one is a series of, is a whole field of research that sort of began in the 1990s called the Adverse Childhood Experiences research. And what that research did was really looked at a number of, let's say, the most common types of adversities that young people can experience before the age of 18. And since the 90s, there's been a ton of studies linking these different adversities to just about any negative health outcome that you can imagine in terms of increase of relative risk. They generally fall into three categories: abuse, neglect, and household dysfunction. And so you can see what kind of falls into here on your own, but also there's things that aren't here that would also constitute adversity. So for example, in remote First Nations, you may have crowded or inadequate housing. You may have environmental disaster that you're responding to every year in terms of evacuations. There may be the experience of racism for some people. For others in the world, there may be exposure to violence and war and other adversities in addition. And so generally, one of the ways I tend to try and think about this is that an adverse child experience might be defined as like that thing which overwhelms in a developmentally inappropriate way an individual's capacity to cope adaptively, such that it produces an environment of toxic stress, probably measured by cortisol response for a prolonged period of time, and has some type of developmental sequelae. And so if you can begin to specify a little bit, then you can decide what goes into a bucket and what comes out of a bucket. And the nice thing about the specificity is then you can begin to think about what to do about that. And so when I learned about this literature, I thought to myself, well, there's a lot of things here that I think are among some of the common experiences that I understand from many of our communities. And so I began looking at this in terms of what this means for Indigenous people. Now there's two considerations here, population level data versus individual level prediction. So it's very, very common for people to look at ACEs, as I just showed them, and think, oh, I wonder how many of these that I have, right? Or I wonder how many of these someone I know has. And then think, well, but I'm not living with these types of difficulties other people have, so what is that? What is that? Well, I think the challenge is you're trying to make individual level prediction based on population level data. And the population level, we're very, very good at making risk predictions, but at the individual level, all bets tend to be off. And this goes across most fields of prediction, at least in mental health. So that's the first part. So let's not try to make individual level prediction on population level data. The second part is people tend to find this area of research a little bit depressing, in that that as the increase in adverse experiences, as that increases, so too does the relative risk of a whole bunch of different negative outcomes in just about anything measured, everything from obesity and diabetes and cancer and addiction and mental health and just about everything. And that's also in a dose-response relationship, such that 2 ACEs incurs more risk than 1, and 3 more than 2, and 4 more than 3. And a lot of people will see that as quite depressing. I tend to see it as a bit hopeful, and the reason why I see it as a little bit hopeful is because of the nature of bidirectional relationships. That is, if 3 ACEs incurs more risk than 2 and 4 more than 3, then the opposite's also true. So 3 ACEs incurs less risk than 4, 2 less risk than 3, and so on. And that's very good, because it gives us a bit of a sense of what we ought to do about it, and we ought to prevent these types of experiences in children. And that's something we can all agree upon. The other thing is that this is something that a lot of us do already, but we just don't really have a framework for thinking about why it's so important and characterizing what it is that we're doing, but also emphasizing the importance more at a public level. And so any time mother has a new baby and is experiencing some type of postpartum mood condition, and her sister or her mother or her cousin or somebody comes to live in the home and help with that adjustment period to ensure that everything's taken care of, you're preventing ACEs, right? Anytime parents go to stay with grandparents for a weekend because there's a wedding and there's going to be alcohol use or some other thing going on, you're preventing ACEs. And so people do this all the time, and you can think in our own communities how we've got even cultural expectations within our families or whatnot when it comes to responsibilities, and this provides a bit of an organizing framework. But ACEs in Indigenous populations is one of those things that tends to not be as well understood or well studied, and it was so something that we wanted to begin to learn more and more about. So at this point, like in terms of my own research program, there's kind of a lot of different things that I do in relation to this, but I'd like to kind of focus on one sort of series of studies that we've been doing, just to give a bit of a sense. And so we published a paper. We looked at available data in Canada from Stats Canada and other sources that looked at rates of adversities in Canadian populations, and in general, on average, the rates of adverse child experiences is decreasing among the Canadian populations. That's good news, except for in Indigenous communities, they tend to be increasing. And there's a couple things that we need to think about when it comes to that. The first is that, well, if we think that there's services that are inadequate now, and we know that at a population level there's an increase of risk of a whole bunch of fairly predictable negative outcomes, then if we extend that timeline out 5 or 10 years, then we're really going to have inadequate resources. And you can do something about that, right? So rather than just letting that happen, you can kind of watch it and begin to deal with that problem. Similarly, across general population, we got very, very familiar with modeling through the COVID-19 emergency. And then we decided, well, we don't need modeling for anything anymore, and we just stopped doing it. But you could be modeling like a tremendous amount of things and being able to make population level predictions way down the line. So for example, in the 90s, we laid off nurses, and now we need more. And we didn't have enough long-term care housing, and we knew that was coming for 60 years. And I run an autism diagnostic clinic, and I assess children who are going to need supportive housing when they turn 18. And I know who they all are already, and I also know that when they turn 18, those resources probably aren't going to be there. And so it's a challenge because those timelines are extended well beyond, say, typical governmental decision-making timelines and institutional timelines, but they are the real timelines that we're working on. And there's a lot of wisdom in orienting our thought in that way. So there's cultural teachings. You may have heard of like a 7 generations teaching, and that we're not sort of meant to think about ourselves as discrete individuals at any one place in time, but rather we're individuals who come from a line of individuals who are responsible to, but we're also responsible to the line of individuals who are coming behind us or after us. And so unfortunately, that doesn't really fit very well with election cycles and other things, but the reality of the situation is this is one way of thinking about how to deal with larger societal issues, is by using Indigenous wisdom and teachings to begin to orient ourselves to like the longer term project that we ought to be collaborating on. And so we were really quite curious about how this works, and in partnership with our organizational partners in the Thunder Bay area, which are comprised of, so it's an organization that I work clinically in, but we also have a program of research embedded in. It's governed by 13 First Nations in the Robinson Superior Treaty area. You can see it here, including my own First Nation. And it's got mandates in child welfare, mental health and addiction, and also health. And so we respond to questions from community. So community questions might say something like, well, we see a lot of trauma going on in our communities. A lot of people, a lot of adults are in need of some type of mental health support or addiction treatment support. So how come this is the case? And so in that way, those initial questions and conversations begin to be had, and then clinicians and researchers and my graduate students and everybody begin to kind of put ideas together as to how we can begin to look at these things. And one of the things that we try to do is understand ACEs more in adults who are in treatment for substance use difficulties. And so what we did was we embedded a study in a treatment setting, and we've been running it now for a few years. And across those years, we're learning a few things. Part of the reason why we started here was observations I was making clinical. I was sitting down in the room with people who had experienced 6, 7, 8, 9, 10 out of 10 of those things that I showed you before. Tremendously traumatized through childhood. In fact, when we looked at the data, this is data from now, maybe a year and a half ago. We've been collecting since, but the average ACE score in this group of individuals is 5.25. The average ACE score in the general population is like 1.2, something like this, right? So you're talking about quite a lot, quite a big difference in terms of individuals here versus individuals just kind of maybe among us. The other thing that we did was we were able to begin to look more closely at different symptom presentations and the predictive power of adversity in terms of determining, again, that specificity for targets. So for example, we saw that ACE scores predicts depressive symptoms, for example, quite reliably. We also found that ACE scores predict utilizing substances to cope with negative emotions. So again, you get another level of specificity. So not only do you have depressive symptomology that's a target, you also have the reasons by which people are engaging in substance behaviors as a target. We also looked at type of adverse childhood experiences in relation to some outcomes. This is suicide-related behaviors. These are people who have attempted to end their life, and what you can see is that odds ratios aren't all sort of even depending upon the type of adversity that people have experienced. And so what that means is, again, another level of specificity when it comes to targeting the specific treatments that we're going to be aiming at those types of experiences. So I talked about relationships in general before, and what I'm hoping, I'm trying to do is paint a bit of a picture or begin to draw in that, you can take a broad model like residential schools leads to intergenerational trauma, but then you can begin to specify what that is and what the mechanisms are. And one of the mechanisms that I think is pretty important, what we're demonstrating in our research is that those experiences that may fall under that broader umbrella of the colonial imposition do something really, really very, very difficult to kind of think about. But one of the things that they do is they disrupt attachment. So biological attachment is a developmental process by which, I mean, not just humans, but any animal that you study experiences that biological process. It disrupts that. Attachment is a very, very important process because it also mediates neurodevelopment. And it mediates neurodevelopment in very, very specific ways. So it mediates limbic type development, so your ability to respond to stress and respond in stressful situations. But also it affects prefrontal cortical development, and your prefrontal cortex is that part right between our eyes, behind our eyes, and it's home to some very, very important clinical constructs called executive functions. And executive functions are sort of the things that we need to plan our behaviors, organize our behaviors, control impulses, respond differentially to novel situations, regulate our emotions, engage our working memory, and so on. In fact, it's places where there's challenges in capacity for people who are experiencing mental health and addiction difficulties. One of the reasons why I like executive functions in general is because I like to choose constructs that are more cross-cultural, let's say. So rather than thinking in diagnostic categories that are culturally bound, we can think about those things that we all have in common. So regardless of what your race or ethnicity or otherwise might be, presumably at some point you have to plan your behavior and you have to organize the things that you need for that, and you have to manage your impulses and you have to control your emotional response and so on. And so they become rather transdiagnostic type factors that are also less culturally bound, and they're tied to very, very important neurodevelopmental mechanisms that we understand. So for example, we looked at the different types of executive functions and found that family substance use, for example, is pretty important in terms of working memory, people's attentional capacities, people's ability to be flexible in terms of their cognitive processes, but also emotion regulation. And so we get back to that initial story I told, right? So we've got evidence-based interventions from a psychological perspective for these different difficulties that people may have in executive function, for dealing with, say, acute trauma type experiences and symptoms related to that. And there's robust literatures that show that there's very, very good evidence-based treatments for this. But one of the things that really strikes me about my own sort of experience in the story I told at the beginning was that, regardless of culture, or even educational background or knowledge or experience in treating things, I had a very, very effective treatment for anxiety that was evidence-based, but it came from a cultural perspective. And so over the years, that has had me really advocating and thinking a lot about how do you bring the very, very best of psychological intervention and the very, very best of culture-based interventions together to be most effective. And the reason why I think that I work on that is because it's an important aspect of identity development, particularly among people who have had their identities undermined as a consequence of, in the case of Indigenous peoples, colonization that's happened for many, many years. We know this implicitly in psychological treatments. So for example, in CBT, you have schema, which is sort of the question of, who do you think you are? And in DBT, we have your life worth living goal, and that's kind of like, who do you think you are? But we also know this in medicine, right? So if you have an athlete who's injured, she goes to her physician and she does whatever that physician says, and she goes to the surgeon and does whatever the surgeon says, and goes to the physiotherapist and does the pre-surgical physiotherapy exactly, and then does everything that the surgeon says afterward for recovery, including all the physiotherapy. And why? Well, because she's going to get back on the track or back in the pool or, you know, back on the ice rink as fast as possible. It's who she is. And we've seen this as well. You know, you've got grandparents who smoked for 30 years, tried quitting, but it never really worked. And then they quit because grandkids came along, right? It's who they are. We know this implicitly in many, many areas of health intervention generally, that, you know, the imaging, the diagnostics, and those are essential parts, right? But behavior change and engaging in the appropriate behaviors that need to be engaged in order to kind of have the best pathway through any type of illness, whether that's mental health and addiction or whether it's other health difficulties, necessarily involves a little bit about who we are, maybe a lot about who we are. And so that's something that Indigenous people explicitly talk about in healing. So if you've ever engaged in a sweat lodge or you know, an origin, you know, you've had teachings about an origin story of where you come from, it's explicit. It's like, this is who you are, this is where you came from, these are what your responsibilities are, and this is who you can be if you do all of these things, right? It's a different way of thinking about the foundation by which people can build out wellness. And so if you can bring these technologies, let's say more broadly together, then you can have more effective interventions. And so what we've done more recently is designed interventions and began testing interventions with Indigenous populations that bring together the very, very best of psychological intervention, but also culture-based interventions. And in those ways, we partner with cultural teachers and elders to do that part, while the psychology people do the psychology part. And really what it is it's in finding the mechanisms of action for intervention. What is it in the intervention that works, say, for people? But also finding approaches that share the same value structure and the same desired outcomes. And in that way, the idea then is to find pathways to wellness that allow us to leverage those resources, those evidence-based resources from a psychological perspective, but importantly, that bring together and reconcile, let's say, and hold as equal the healing practices of the other many other cultures across Canada. So with that, I think I will say Chimi Gwech again for the opportunity to be here. I very, very much am honored and very, very thankful for your attention today. So.
Good morning. My name is Shahzad Iqbal. I'm the Country Medical Director for Moderna Canada, and it is my distinct pleasure and privilege to introduce the next speaker, Dr. Jose Belezan, the celebrated recipient of the 2023 John Dirks Canada Gairdner Global Health Award. Dr. Belezan will share his insights on calcium for preventing hypertension during pregnancy, the research journey from observation to population-level implementation. Dr. Belezan is not just an obstetrician, he is a trailblazer in his field. His academic journey began at Rosario University, where he trained as a medical doctor before earning his PhD in biology of reproduction from the prestigious Salvador University in Buenos Aires. Now he's making waves as the principal investigator in the Department of Research in Maternal and Child Health at the Institute for Clinical Effectiveness in Argentina. In addition to his primary role, Dr. Belezan holds esteemed positions as an associate professor at both Tulane University and the University of North Carolina at Chapel Hill in the United States. He also contributes his expertise as a senior researcher at Argentina's National Council for Scientific and Technical Research. Please join me in extending a warm welcome to Dr. Belezan as he takes the stage.
Okay, hello. We're moving from basic research, clinical research, psychiatry research, and now global. Okay, let's see what. I can say what it's an honor to be here. Thank you, everybody, to come. Yes, I am a obstetrician, I'm a medical doctor. I did practice, take care of pregnant women and the baby for many, many years. So my talk will be related to obstetrics, pregnant women. So one thing that important disease what we face taking care of pregnant women are what we call hypertensive disorders of pregnancy. We have different classifications, such as hypertension, preeclampsia, and the most severe status is eclampsia. We're very worried about that because it's the principal obstetric cause of maternal death around the world. And then it also worries us because there are around 50 to 70,000 women die in the world due to this complication of pregnancy. And I can't say more words about the tragedy of maternal death. Sorry to bore you in this, but maternal mortality reflects the most striking global inequities in health. In the world least developed countries, maternal mortality rate is more than 40 to 60 times higher than for high-income countries. Inequality, a great inequality. I needed to say maternal death is a tragedy, involves the possibility that their child, 46 more times increase the risk of neonatal death of the baby, 15 times more increased risk of child death in the first year of life. The surviving children have exacerbated surviving children's vulnerability to illness and malnutrition, short underage education, too early labor participation, too young marriage and parenthood. You see the consequences and why you are so worried about the tragedy of maternal death and about this disease. Not only mothers, it has been estimated that the hypertensive disorders of pregnancy precede about 1.5 to 2 million deaths annually. So you can see we are facing a quite severe complication of pregnancy. But in my history, I have the possibility to go to work to Guatemala in Central America. And I work in the Institute of Nutrition. And there is a peculiarity in the diet of women, extremely poor women, descendant of the Mayan indigenous population in Guatemala. And it's the staple food of these women are corn. The poor they are, most is the diet involved corn. The most poor, 90% of diet is the corn. And how they treat the corn? It's a Mayan custom for many, many years. They put the corn in lime water. They put a chocolate lime in the water, and then they leave the corn all over the night, and the next day they carry the corn to the mill. And the consequences of this is that, are you familiar with the tortillas of the women? The consequence is that the tortillas have a very high concentration of calcium, and it's in a institute that demonstrates that this calcium is as good as the calcium from skim milk. So it's a calcium of high quality because of the aggregate of lime in the water. So this is where is here? We did this rough epidemiological studies, and we put here the intake of calcium, and the most severe condition is the eclampsia incidence. And then as you see, the countries with a low calcium intake have higher incidence of preeclampsia, but this does surprise us because those are low-income countries, the majority, and low-income countries are worse attendance of pregnancy. But there are two countries here that not belong to the low-income countries. One of them is Guatemala, that for this peculiarity I mentioned you, have a high calcium intake and low prevalence of eclampsia. But there's another country, Ethiopia here. What's Ethiopia doing here? He must be there. But also, there is a peculiarity in Ethiopia that have a high calcium intake and low incidence of eclampsia. And what's happening in Ethiopia? The main staple food is a grain called teff, T-E-F-F. And teff has a very high concentration of calcium. So very poor women in 2 countries having a low diet, but having a high intake of calcium, have low prevalence of the disease. Then we wonder, could be any relationship between high calcium intake and the development of the disease? And we started a series of study to see, first of all, we reviewed the literature to know this was in 1977. We don't have internet and so on, then papers. But we didn't find anything, just only one study done, published in 1976. It's say that persons living in villages along the banks of the river in New Guinea, calcium content of river decreases as the river flows downstream, while blood pressure of the villagers living along the rivers increase. That's the only thing we can find a relationship between calcium intake and blood pressure. Then we start to do a serious study. The first study was done in rats, and we submit rats to a calcium-free diet, no calcium in the diet, and a normal diet. And when we saw that the rats with a calcium-free diet, no calcium in the diet, increased the blood pressure significantly higher than the rats with a normal diet. That was the first observation and direct relationship between calcium intake and blood pressure in animals. Then we move to you students that are volunteer. We can restrict calcium in the diet, those who decide to give 1 gram of calcium per day of a sample of young people. And what we saw is those young people receiving a supplement of calcium, the one receiving a placebo doesn't change. And those receiving a calcium supplementation decrease their blood pressure value. A direct relationship between calcium intake and blood pressure in young people. And we did the same in pregnant women. For pregnant women, that was a placebo control group, and those receiving a supplement of calcium, and then there are a reduction in blood pressure in those women. Then we were quite surprised that the initial hypothesis have an explanation about the reduction of blood pressure with calcium supplementation. We always deal with, you researchers, about mechanisms involved on that. What could be the mechanism regarding the relationship between calcium intake and blood pressure? And then we did have a hypothesis of these different steps are well known. There is a step that when you reduce calcium intake, there is an increase in parathormone levels. There is also shown that the increase in parathormone levels increase intracellular calcium. When the intracellular calcium increase in the vascular smooth muscle, increase the muscle reactivity and an increase in blood pressure. So that's the hypothesis of the explanation. Then we did study, okay, could be all of this mediated by parathormone level. And then we work, to remember the previous one, low calcium diet, high blood pressure. And then we suppress, there's animals parathyroidectomized, animals that took away the parasite gland, and we compare them with animals in sham operation. And then what we found that the decrease in calcium intake, not having parathormone, parathyroid, doesn't increase blood pressure that we saw in entire animals. So we think that the mechanism is mediated then by parathormone and the change in intracellular calcium. Then, quite recently, a colleague working with us I don't know if you're familiar with the Cochrane Review. The Cochrane Review is an initiative that joined studies doing the similar studies, and then they have a compendium of what happened in this study. And then, yes, there is a decrease in blood pressure in individuals with calcium supplementation. Then what we did, after I showed these studies in animals, then we went to women. And we did a randomized clinical trial published in 1991, where women at 20 weeks, they were submitted or volunteered to receive a placebo or to receive calcium, elemental calcium around 2 grams per day. And they followed these women all their pregnancy. And when we found that there was a reduction, about 37%, on women who received calcium in comparison with women who received a placebo. One important thing in clinical work is the replication of the study. Unfortunately, our study was replicated in 27 studies involving around 20 this amount of women, doing exactly the same, giving some women placebo and some women a supplement of calcium. And this study found that there's a nearly a reduction of half of preeclampsia of this disease on women receiving calcium in comparison with a placebo. And the author of this, we call it meta-analysis, look what happened in women who have a low calcium intake before pregnancy in comparison. And then what we find is that women having a low calcium intake, the reduction is greater, 64 reduction in preeclampsia. After this, with this author, we did a study in Africa and Latin America, and we give women before pregnancy a supplement of calcium. And during pregnancy, all of them receive calcium because it would be unethical not to give it. And in these women, there is a further reduction of 34% of preeclampsia when we supplement them previously to pregnancy. So there we are. Another thing I would like to show you is that in the Cochrane review, in the supplement, in 11 trials, 15,000 women, there is a 24 reduction of preterm birth. You know, preterm birth is something that is really an important problem all over the world, not only in low-income countries, but also in high-income countries. So I don't know the mechanism, but we saw, the Cochrane review saw a reduction of preterm. So fortunately, I know you're familiar with WHO recommendations. WHO recommendations are very strict recommendations. They put a number of experts analyzing all the evidence, and then after paper joining many times, they make the recommendations. And currently, there is a strong recommendation by WHO saying that in areas where dietary calcium intake is low, calcium supplementation during pregnancy is recommended for the prevention of preeclampsia in all women. So every woman in countries with low calcium intake should receive a supplement of calcium. We feel very proud about this, that from this initial observation involved a strong recommendation of WHO. Then one thing that analyzing maternal death by obstetric causes for more than 30 years, what we saw is that there was a great decrease of maternal death due to hemorrhage. And there is a very simple procedure to prevent maternal hemorrhage. It's just a puncture of oxytocin in the delivery, and then this prevention, maternal death by hemorrhage can prevent it. But what happened with preeclampsia? This is stagnant all over 30 years. There are not being major reduction in maternal death due to preeclampsia in this year. So that's why we are so worried about the possibility to implement the recommendation of improving calcium intake to prevent a disease that causes maternal death and doesn't make any variation around the world. Let me show you another result of our studies. The same women that I showed you that were randomized to receive calcium and placebo, where we did a follow-up of their children. We followed their children around 7 years old. It's not easy to do follow-up of women, but we managed to recruit children at 7 years old and to measure blood pressure to them. And when we find the following, those children whose mother received calcium supplement during pregnancy are around 7 years old, show lower blood pressure values above the 95th percentile than the children receiving a placebo. That means a reduction on the children due to the diet of the mother during pregnancy. We don't know how, we don't know the mechanism, but there's some kind of imprinting of the fetus regarding maternal diet. And that's an important message derived from these studies. There are not too much studies doing the same, supplementing calcium during pregnancy and measuring blood pressure of children, but the meta-analysis of the few studies show a reduction of blood pressure on children whose mother received calcium supplementation during pregnancy. Let me show you something peculiar. When we did the study, mother told us, you know, my children have better dentition than my other children. But the women were blinded. They don't know what they were assigned. Okay, let's see if they're right. And we did a study at 12 years old of the children, and the children whose mother received calcium have much less number of decay, miss, and filled teeth. There was an effect on the dental process of the children regarding the calcium intake of the mother. What curiosity. We don't know the mechanism. We don't know what happened. We're thinking epigenetic factor, but we don't have. Then how about following further this progeny? It's not easy because for following 20, 30 years, the researcher wouldn't be there for the year. Then we work on laboratory animals. And we did the following study. Virgin rats were assigned to differential calcium diet. A group of them received a normal calcium diet. A group of them received a low calcium diet during a period of time, and other group with double calcium diet. After that, we have a couple, and then they became pregnant, and then this is the period which there are women before pregnancy and during pregnancy receiving just a double calcium take, normal and low. And then after that, we follow their children, no. And then we follow them for 52 weeks. 52 weeks is the span of the life of the rat. No? And then we follow them for all the span of their life. Then what we found it's clear, no, the methodology. Then what we found is quite surprising because we found that the progeny of rats whose mother received a normal calcium diet, this is the values, and the one who received a low maternal calcium, there was a strong difference, 12 millimeters of mercury of the progeny of the rats receiving a diet with low calcium content. Then that's a validation of what we found in human beings or in animals, laboratory animals. But let's go now to the world, and let's to see inequities in the world. And what happened about diet of calcium in different situations. And we did this study. We collect 150 studies, this amount of women in 37 countries, that have measured calcium intake during pregnancy. And what we found everything that in high-income countries have a good calcium intake because around 1 gram per day is a good diet. But it's lower in low and middle-income countries, and 82% have values below what is desirable. So the issue is not intake of calcium. The issue is availability of calcium. There are many countries, the great majority, here is North America, here are you, but the vast majority of countries have lower availability of calcium intake. And it's calculated that around 3.5 billion people are at risk of inadequate calcium intake. So the issue is to say, okay, we have something that could be applied, must or should be applied in low and middle income countries where the diet is insufficient, but how we can manage this? It's not easy to have a good antenatal care in low and middle income countries and to give pills to every woman, but it's intervention that we want to promote. But we are looking for other things, and in this recommendation, WHO, they mention about dietary counseling in all women who are considering pregnancy. I'm sorry. But what we say here is that food fortification of staple foods with calcium may be an important public health intervention. And we agree this. We agree that if we can fortify staple foods, women can achieve a good calcium intake before and during pregnancy and prevent them from this severe disease. And we are working on this because calcium supplementation also, I show you, reduce the risk of hyperpression of children born to mothers supplementing during pregnancy, or they show that. And also have better cholesterol levels and decrease the risk of colorectal cancer, adenomas. I don't know why, but it has shown that. Those has many benefits besides the benefit I mentioned about blood pressure value. Also improve bone health. Then how to manage this problem worldwide? One of them is having a pill that we can provide this pill to every woman, but probably you know that every pregnant woman in lower income countries should receive iron. The anemia in lower income countries is a major problem. And then it could be feasible to have a poly pill, and the poly pill will have iron, folic acid, micronutrients, and of course calcium. No? And the pill could give in one per day. That's an idea. We didn't work on that. We are trying to work on this possibility. The other thing is we're thinking in the fortification of possibility to fortify water or flour. These are simulation studies. These are values that we have on diet in different countries. And then what we did, is simulate an increase of 500 milligrams of calcium per day. And unfortunately, very few persons will be above the upper limit of recommendation. So what we think is that the possibility to increase calcium intake at population level won't involve people who have upper limit that could be associated with damage. And we're thinking of water. Water is universally consumed. However, most drinking waters have low calcium intake. And when thinking the possibility to supplement or fortify bottled water and also to fortify pipe waters. And what I'm showing you here, it's a study of taste. We assign a group of women and they taste different concentrations of water with calcium blinded. And we found that the limit in bottled water is around 521 milligrams. Let's say that below that, the group of people doesn't perceive that there's something different. And then we can supplement with carbonate calcium water with figures above or around 500 milligrams. What happened with pipe water? The regulation is that pipe water should be, couldn't be above 120 milligrams per liter because it could put problem in the tubes. But and the pipe water we use calcium chloride, and also the taste, when they perceive a difference, is around 166 milligrams per liter. So it's upper than the limit imposed by regulation. So there is a feasibility to fortify water, and the people won't have a different taste in the water they consume. And what about flower fortification? There is many experiences about flower fortification in the world, but only one country in the world have mandatory calcium fortification, and it's UK. And the calcium fortification in UK, let me see with that. Yes, mandatory calcium fortification in UK starting in the war, 1943, and remained there for such a long period of time. And you know how UK scientists evaluate what's going on, and they say, shall we stop the calcium fortification or proceed for calcium fortification of flower? And the consideration that they are going to proceed. That's consideration done in 2013. And they say that they have health benefits. Calcium intake will be affected in all ages. It's a question of health equity, because the impact will be greater in low economic groups, increase the cost of social care and NHS. They say that fortification is not burdensome from the industry. And some curious things that the premise cost is probably cheaper than the equivalent weight in flour. The premise to add calcium could be cheaper than the flour involved in the same volume. So there we are, we're working on all this kind of research, from basic research trying to explore mechanisms of the imprinting of the fetus, and see how we can universally apply a benefit to mothers and their children. I put out some of what we show. In some way, there is observation of an ancient Mayan tradition in Central America led to the hypothesis. The corroboration of the hypothesis to the discovery and intervention that can save maternal and baby lives. This intervention will also involve benefits on the progeny. Intervention to increase calcium intake and range from calcium supplementation before and during pregnancy, and the fortification of staple foods like flours, rice, and water fortifications. The advances in these interventions will aim to universally apply to achieve equity in the world. Okay, thank you so much for this opportunity. Thank you.
Our next distinguished speaker, Dr. John Jumper, a respected recipient as well of the 2023 Canada Gairdner International Award. Dr. Jumper will be sharing his insights on the captivating topic, highly accurate protein structure prediction and its applications. In addition to being the AlphaFold lead and senior staff research scientist at Google DeepMind, Dr. Jumper is also a pioneer in the field of protein dynamics. His academic journey began at Vanderbilt University, where he earned a Bachelor of Science in Physics and Mathematics. He then furthered his studies at the University of Cambridge, securing a Master's in Philosophy and Physics. At Google DeepMind, Dr. Jumper is at the forefront of developing innovative methods to apply machine learning to protein biology. His contributions to the field are shaping the future of the biological research that is to come and have the potential to revolutionize our understanding of protein structures. We are truly privileged to have such a distinguished scientist with us here today. Please join me in extending a warm welcome to Dr. John Jumper as he steps onto the stage.
It's truly an honor to be here. It's nerve-wracking to follow such talks and such impressive work. And So I was originally trained as a physicist and got my PhD in chemistry. So I'll try and bring us a little closer to the atoms in this talk and really start to think, I think starting at the incredible manufacturing process of the cell and what evolution has created. And we talk so much about our 1D genome and this line on which all these bases are written. And these are the instructions for life, and especially the instructions for the nanomachines within our cell, the proteins. And this solves an enormous problem of how do we create rich, complex 3D objects, the kind of machines that we need to do the business of the cell. How do we write them in a single line? And the answer turns out to be that we, within our DNA, are encoded many, many instructions for building proteins. And those instructions, of course, are regulated and turned on and off. But they very simply say, well, you take the 3 bases of DNA, roughly. Apologies to all the biologists in the audience, but for the non-biologists. And you have one single protein amino acid that is added to the chain. And you build this chain one at a time like beads on a string. And it's a quite elegant solution, and there are 20 types of beads, and I'll show you a picture on the next slide. They're just made of atoms. They're quite simple. But the magic of this is that some of them, for example, are more greasy and want to get away from water. Some of them like water. Some of them are charged. And so they create a complex pattern of interactions which just via physics is able to fold up itself. What Anfinsen notices is that this is encoded in the protein sequence what structure it should have and what kind of rich 3D configuration that's able to do everything from what pulls when our muscles contract to making all the products to breaking down our food. All of these machines are encoded quite simply into our DNA. And so the building blocks here, the 20 amino acids, they're very simple. They're small numbers. You can see the atoms here, and yet they give us the diversity of biology. And they do that in part because they assemble into quite large structures. So hundreds of amino acids are attached into a long line, and you can see the ball and stick atomic representation on the left. And you probably, even trained protein biologists, will have trouble tracing the individual chain through that molecule, except they'll probably all know which kinase that is. But if you look in the middle, we have a lovely cartoon representation by Jane Richardson and others that gives us a nice, easy to trace path through here and kind of lets us understand how this protein is organized. And the real question that we've had for a long time is, as we learn more and more about our DNA, as we get more and more instructions for proteins, well, what do they look like when they're built? What is the 3D structure that we see in the middle? How can we figure this out from simply what we can find in DNA? And this is really important because DNA doesn't tell us the whole story, that we really need to assemble the machine. So this is a minimal cell, right? Intentionally constructed to be one of the simplest genomes that you can have. And you can see it's incredibly densely packed with these proteins in different colors, as well as DNA strands and others. And you can see it's an extremely dense environment with lots of interactions, that there's lots going on. That this is the crowded factory, if proteins are the machines, then this is the crowded factory floor in which the business is done. And each of those individual little colored blobs are really, really worthy proteins that have often been studied for the entire career of biologists and really contributed to our understanding of health and disease. But how are we able to step back? How are we able to build the kind of tools we need to understand the larger complexity and to complete this picture? And so scientists have worked for many years to find the structure of proteins. And one thing to think about is that proteins are very small, right? Their wavelength of light is about, say, 400 nanometers. The size of a protein is a couple nanometers. So these proteins are much, much smaller than light. You won't see them directly on a microscope. You need something with much, much shorter wavelengths, which means much, much brighter light. And so what scientists have figured out, and there are a couple of techniques to find the structure of proteins, but they're all incredibly difficult. So the method of X-ray crystallography is that you first convince these proteins to form an ordered crystal like table salt. That's quite a bit of work. That's no repeatable recipe. It often takes years to do this first step. Then you get to take it to an enormously large scientific instrument. If you look carefully, you can see individual cars here, right? So we take it to an absolutely enormous instrument to get very, very bright X-rays, a synchrotron. And then we take an image of this protein crystal. And then once we have that, we can solve a rather quite difficult mathematical problem, which sometimes fails, phasing for the experts. And then you can finally get a single structure of a protein. And this has been, over about 70 years, developed and refined, but still intensely difficult. And through enormous societal investment, we've determined and deposited about 200,000 protein structures. But we've determined billions of protein sequences, right? We've had the sequencing revolution. We find protein sequences 3,000 times faster than we determine protein structures. And for example, even only about 1/4 of the human proteome in terms of residues has been covered by an experimental structure. Now, the other thing is that there was quite a bit of foresight in this field. And these data are very valuable, and they were aggregated into the Protein Data Bank over 50 years. And in essence, this represents the full output of the structural biology community. You essentially can't publish a paper on a protein structure without depositing it. And this leads to an incredibly diverse and exciting set of really, if you ask machine learners, incredibly clean biological data. Of course, if you ask experimental biologists, they'll tell you about all the things that are wrong with the particular crystal structure that they last looked at. But this creates an incredibly important problem, and really a really interesting one from a machine learning point of view, because what we're talking about is not machine learning to, say, replicate human judgment or do something that would take humans a second or a minute or an hour, but this is something that the best scientists, that people who will get their PhDs will take a year or years, each one. And it creates this really important problem to think about: how can we use machine learning? Can we use machine learning? I'm giving the Gairdner lecture, so the answer is yes. And really what we've created is, in a sense, a very simple system, at least conceptually. We take the thing on the left and we make the thing on the right. So the left is a typical protein sequence, 1 letter per amino acid. Each letter is an individual amino acid. We put it through a computational system, and I'll tell you a bit about how we built this. And on the right, you can see one of our predictions. This was a blind prediction. We didn't know the answer at the time we made it. The dark blue is the prediction. The green is the experimentally determined structure. So you can see an extraordinarily, as Dimas talked about, angstrom level, extremely close correspondence between the prediction and the experimental result. And that's really important, because often it's very small parts of this structure in which the experimental biologist needs to make the hypotheses to perform their experiment. Now, I'll show you a couple more examples. We didn't just do this once. We can, of course, do this many times. And you can see some really large and intricate structures that can be predicted computationally. In fact, the one in the middle, we could both predict blindly with the latest version of our software, but also parts of it used AlphaFold to determine the structure, so it's kind of AlphaFold on both sides of that one. But we can really find these intricate and beautiful machines that do all sorts of functions within the cell, and we can understand them. Okay, so how did we do this? And if I showed you this and I talked you through this, it would be, very exciting for certain audiences, but, you'll have to see one of my other talks for that. And I think it's really, looking at the details of all these components and their mechanisms and everything else is kind of the wrong way to think about it for a reason I'll make an argument for later. I instead want to kind of take you through the conceptual thoughts that get us there. And the first thing is a story that is before us, and some insights in the field. First of all, kind of telling the story of causality. We have the protein sequence on the left, represented as kind of lollipops. And then because of physics, that causes the protein to fold up and have a structure. Now that structure has a very strong influence on evolution. These structures take a long time to evolve. They change slowly. So if you look at many copies of the same protein, say the analogous protein in mouse and in human and in dolphin and in yeast, you will often see very similar structures. And so this gives us a new source of information, and it's kind of been talked about in some narrow ways with terms like coevolution and profiles, but I think you should simply say that, well, that structure had a really strong influence on evolution, and we can measure evolution really well because we have all these sequenced genomes. So when we try and say what is the structure of this protein, we'll not just use the sequence of the protein, but we'll use the sequence of many related proteins in order to put into our predictor. That alone is not enough, and there have been a lot of systems built on coevolution, and certainly helps, but it doesn't solve the problem. And these systems also use machine learning, and deep learning. And I think one thing I'll just say here is that deep learning is extraordinarily generic. Demis talked about so many incredible applications. And there are a couple of standard gadgets and ideas in the field that have typically been developed in the context of text or in the context of understanding images. And these can often come with proofs. They can do anything. If you give them enough data, they can learn about any function. But that's not so good, because we don't have web-scale internet data. We have those 200,000 incredibly precious structures provided to us by the whole of the structural biology community, and we need to make really good use. We need to really, instead of taking something that can learn anything and show it our data and hope for the best, we need to figure out how to rebuild deep learning for proteins, for the unique facts. How do we build the intuition that scientists over the years have developed about proteins in exactly the right way and build it into our learning system so that we kind of have an equal conversation between the data, which comes from experiment, and the kind of ideas that come from theory and our own understanding, and how do we fuse those into a deep learning system at the most core level? How do we build new building blocks to do that? And so we came up with a number of ways in which we built these insights into the network structure, and for the interest of time, I won't really talk about them in detail. But it's basically rewiring the components so that the things talk to each other in meaningful ways, so that we exchange information between the evolution and the physical representations in relatively precise ways with a lot of experimentation to get this right. And we developed the ideas on how to build what we would consider an end-to-end system. So previous systems, including our AlphaFold 1 system, had made predictions about the protein. Certain residues might be close, et cetera, but they didn't produce a protein structure. And that meant that you got all these answers, and then you put it into another very complex system to try and get a structure out of it. And what we found instead is that through a set of ideas, we could simply have a network that was accurate as previous systems, but produced a protein structure immediately. And so we could get, obviously the system was wrong at the beginning, but we could get, 100 wrong answers a minute, right? And that lets you improve, that lets you change. It's really, really important in a deep learning system that you produce the output that you want to improve, that you have a measure of what will take you closer to the desired output. We also had, using this funny machine learning word of inductive bias, but we had ways in which we kind of bias the system to learn about protein physics and geometry. And you can think of this, for example, for experimentalists in the group who have fit their data with a sigmoid, because there's good reasons to believe that your data comes up and saturates. That's much better than fitting it with just a high-degree polynomial. You want to fit it with functions that make sense and are aligned with your understanding of the underlying physics and biology and geometry of the problem. And so we also de-emphasized really what you knew at the beginning, the spatial chain, or the chain structure, the sequence of amino acids, because as a protein folds, the primary sequence becomes less important. That you have a piece here and it goes off and the chain does many, many things, and then it comes back and another piece comes back. It comes here right next to it. And what's really important is that maybe residue 100 and residue 300 are extremely close in space. And so as soon as the network kind of figures that out, to use an overly anthropomorphic term, you want it to integrate that knowledge into all further processing. And so we developed ways in which, in essence, and as Demis showed on the iterative development of structure, that as it learned pieces of the structure, it would then use those to communicate, to analyze, define new parts of the structure. And at all at the same time handling uncertainty. Much like scientists, the network never knows anything for sure. And so you must always deal with kind of iteratively building and building on the knowledge that you have. And in essence, all of these processes, we were able to kind of figure out gadgets and ideas within the deep learning framework that kind of enhance them. And what we did is every time that we enhanced this kind of, alignment between our problem and our neural network, we got more efficient with the data we had. So I think one external study had estimated that AlphaFold 2 was about 100 times more effective at learning from protein data than AlphaFold 1, meaning that AlphaFold 2 trained on 1% of the PDB was as accurate as AlphaFold 2 trained on 1% of the PDB was about as accurate as AlphaFold 1 trained on the entire PDB. So as you kind of pre-organize the network to learn about protein structure, you get incredible efficiency improvements. And I won't go into all the details, but these kind of processes are really what drove us to learn infinitely more, because we couldn't wait for the PDB to get 100 times bigger. Now, within that were many, many individual ideas. And I would say, you know, several of these were absolutely brilliant. But if you actually look at them and you're really honest and you perform knockouts on your ideas, which machine learners call ablations, we'll see that each one of these, so we were about maybe in GDT, about 30 GDT better than AlphaFold 1 and other systems. So if you look at, this is AlphaFold 2, and if you look at removing any of these ideas, which each of these individual rows are an idea that's been removed from the system, you can see it has a quite small effect. Now, some of the double knockouts have a much larger effect, where we take out 2 ideas. But ultimately, what we found is that it wasn't just one idea. You really needed philosophy. We needed to build as a team on each other's ideas, and we needed to progress many, many ideas, each of which increased the efficiency a little bit. And I like to say that no idea was worth more than 1 or 2 points on this 100-point scale. So it really required many, many ideas working together in order to reach the goals that we had set for this system. Now, what's also really, really important is I don't think, honestly, that I would be standing on this stage if we had produced a system that was pretty accurate, but no one knew when it was accurate, right? And that wasn't worthy of the trust of experimentalists, because some of its answers are incredible and some of its answers are not so incredible. But we had kind of 2 things that were really, really important. One is that we found actually we could rather directly ask the network when it was right. That we trained an additional output of the network where we knew every answer, of course, that when we were training on PDB, when the network was learning. And so we could measure the error in that answer, and then we could have another part that said, before it got to see the answer, how wrong do you think you are? It's much like taking a test and someone asking, how did you do on the test? And you go, I think I got, maybe I got a C, and I'm pretty sure I didn't get this question. I'm pretty sure I got that question, right? It's not that you know the answer to the question you didn't get, but you know you didn't get it. And so it's a very similar process here, where the network is predicting its own error. And so you can see the orange and yellow and red colors are really indicating areas where it believes you shouldn't trust this answer. That's not a prediction. I had to put the atoms somewhere, I put them there. Whereas the dark blue, I'm really pretty sure about this. And we developed another measure that I won't go into for time, but we developed many measures that help scientists say, okay, now here's my prediction. Now, which parts of it should I use? Which parts are worth weeks or months of experiments? Which parts should I order a new gene because? And we also had the advantage that AlphaFold's answer, when it didn't know, wasn't so hallucinated. It looked for the non-structural biologist, this doesn't look like a protein. This isn't how proteins look in isolation, and so it, in a sense, looked totally implausible when it was highly uncertain, which helped people ignore it. So this was very, very important in terms of applications. I think it's key for machine learning and science that these systems tell you when to trust them, that we develop the measures to do so. Now I'll talk about a few applications, and I'll try and talk just a little bit about some of the non-obvious ones, because I think one of the really special parts of kind of releasing AlphaFold to the world for me is how many different ways it was used off-label. How many different kind of ideas that people tried that weren't just let us predict the structure as we would do crystallography, but finding kind of new uses and ways to do it. Some of the earliest, and there's been some very impactful work on this recently, has been finding that AlphaFold, and a version that we released shortly after called AlphaFold Multimer that predicts protein-protein interaction, is in fact quite useful at understanding, at discovering protein interactions. That in essence, AlphaFold producing a confident structure of 2 proteins is evidence, normally in combination with molecular biology evidence like yeast-2 hybrid or co-IP, is evidence that these proteins actually interact. And so there's been a lot of work just doing what people have started to call virtual pull-downs, where they find new interacting proteins, they discover in some cases very important components of say the DNA replication pathways, and they do it in essence by using this computational tool and by using it across often hundreds or thousands of proteins, which are just totally hopeless to do experimentally through structure determination. And we're starting to really get at the point where we can talk about it. We're certainly not there yet, but really mapping out the protein interactions and the relevant interactions within the cell. Another really interesting thing and another really surprising thing, and I will say one of the disappointments of AlphaFold is it's not very sensitive to destabilizing mutations, and the alpha missense work that Demis talked about really addresses that in a different way, in a different context. But it does know a fair amount about protein physics. And what people found relatively rapidly is people who design new proteins, who are trying to invent proteins, that it was very, very useful to simply ask, AlphaFold, what structure is my design protein? And if it gave the answer that they expected, then they would go off and make this protein. If it gave a different answer, it said it doesn't have a structure, they wouldn't make the protein. And that increases the success rates of protein design in some cases by tenfold and has become a quite standard part of the protein design pipeline, in addition to the ideas inspiring protein design systems. But we're really seeing that this kind of trained experimental system that we really aimed at understanding natural biology is useful in how people design proteins. We're also seeing, I think, a lot of instances now in which AlphaFold is coupled really, really well with orthogonal experimental techniques. And this is a study that I really love on a system called Midnolin, which basically was a quite understudied protein before this study. And they discovered through molecular biology means that it regulates, that it targets for degradation quite a few proteins, but exactly how was not known. And then using AlphaFold, they found that if they took 500 proteins who were degraded when the Midnolin expression was increased, for 200 of them, they could produce a structure that looks like this, where catch 1 and catch 2 are two domains of Midnolin. But this strand in the middle is another protein that's targeted for degradation, and you can see it forms this 5-strand sheet. It's very aptly for the name caught by Midnolin. And in fact, almost all these proteins have this. And if you take, I think they took 5 or 10 of those proteins and said, what happens if we delete this short region? Then they're no longer degraded by the Midnolin pathway. And so we're really have a pretty firm understanding almost immediately in a paper that I think had, would have taken quite a while to work out via traditional structural biology. We have almost immediately a mechanism, and then we have hundreds of examples of exactly what is the motif that is recognized. And we're really kind of skipping steps in the biology, and we're able to start to understand and start to see science get a bit faster. And I think we're going to see that accelerate more and more as both new applications of AlphaFold are discovered, but as new AI systems are developed in biology, and we start unlocking many, many more opportunities. And I think really, as Demis said, AlphaFold should hopefully be seen as the beginning of an era in which we're really using machine learning in partnership with experiment and to amplify experiment. And AlphaFold, and I think we'll see this in a lot of other cases, we started with sparse experimental data where each of these was studied in depth to answer a particular biological question. And there are a lot of pieces missing, and so we were filling these boxes in one at a time. And really through machine learning, we have this opportunity to complete it all at once, that when we reach a critical mass of data, we can convert that into a really complete picture that can be used in all these different and creative ways. And we've converted something from a really, really hard years-long process to instantaneous. And when we accelerate things by that many orders of magnitude, people come up with really creative uses. It really starts to impact how we do biology, and we will almost certainly find new ways to do this kind of trick again in both protein contexts and non-protein contexts. And with that, I want to thank and I want to say very much that this was a team effort and teamwork, and can say by far the best team that I've ever worked with in my career. There were about 15 people that worked on this full time. And really, I want to thank everyone that made AlphaFold possible. The methods, the human proteome, the work that we did in collaboration with IMBL EBI, the CASP community, and most of all, PDB and the experimentalists, some of which in this audience, who had some influence on AlphaFold's parameters. Thank you.
Thank you, John. Thank you, Demis. Thank you, everyone. I think we're finished. Great morning. Wonderful talks from our laureates, our early career investigators, our Momentum awardees. Thank you, the audience, for coming and hearing, and I hope you were inspired. Be back next year. We'll have another set of great talks for you.